All right, everybody. Good morning. I've got 9:01 Central Standard Time. Let's get started with the EHS Academy, our third week of this offering. This is our final week. I've got really good lineup of presentations coming up here shortly, which include biological safety basics. So Dr. Kristen King, who runs our biosafety program, is going to cover biosafety basics. We've got basic ergonomic assessments. Robert Plant from our team is going to go over basic ergonomics. And then we'll conclude with radiation safety basics. So Dr. Janet Gutierrez, who runs our radiation safety program, is going to present that material. So as you all know, this lineup in terms of the topics that are included in our virtual sessions have been strategically organized sequentially, starting with the risk management and insurance basics that we covered in week 1, the fire and life safety and occupational safety basics that we covered. Last week was chemical safety, occupational health programs, and hazardous waste management basics. And then we'll conclude with the topics that I just mentioned here today. We've got the first 15 minutes dedicated to reviewing the asynchronous content from week 2, from last week. That includes the information from the chemical safety basics and the hazardous waste management and environmental management basics that were provided in the asynchronous folder. So Alan Lucas and Dr. Will Bryan are here with us for the next 15 minutes. And so we wanted to open it up for discussion in case you guys have any questions, comments, or just want to have a general discussion about any of the topics related to chemical safety, industrial hygiene, hazardous waste management, or environmental programs. I'll monitor the chat here. If anybody wants to come off of mute, feel free to do so.
Good morning, guys. While we're waiting, since for the chemical safety portion, we didn't get to demonstrate a lot of the instruments. So to supplement this, if you would, you can go to sgsgalson.com. So that's sgsgalson.com. Okay. And then once you're there at that site, you can go to the Resource Center. Okay, it should be right on that front page. And you click that, and then you'll see another link for an instructional video gallery. Okay, and if you click on that video gallery, you'll see videos of basically how to set up active sampling. They'll go in depth on how you calibrate your pumps. Goes into how to use direct read instruments. Pretty much all the instruments that you'll see here on this site is kind of what we do. All the instruments that they use is basically what we use. Some of those videos, they're using older models. So some instruments may have been, may have newer models, but the idea is still the same. But I've found those videos to be quite helpful.
I think it's a great suggestion, Will. Do you mind putting that information in the chat just to make sure everybody has that?
Yeah.
And in addition to what Will just mentioned, for those of you who have the capability, in addition to this online resource, we often dedicate one of our continuing education sessions that we provide internally to our own EHS staff to doing a hands-on review of the industrial hygiene equipment and monitoring capabilities that we have internally within our departments. And so that way, everybody has an understanding of what our capabilities are and how to use some of our basic equipment for things like indoor air quality evaluations, odor complaints, etc. Our entire team is not necessarily expected to be proficient on every device, but it is really helpful for our staff to understand what equipment we have what it's used for, and what our capabilities are for our own in-house sampling capabilities. Great
And
suggestion.
sorry, did everyone get that? Scott, did you see that come up?
Yes, sir.
Okay, I've also added another link, skcinc.com, skcinc.com, and that's where we pretty much order all of our media for IH sampling. And if you go to that website, you'll see at the top of the page, you'll click on sampling guides. And then once you're there, you'll see a pull down. It says OSHA NIOSH sampling. And you click that, and then you'll see some spaces for, inputting of a chemical, an OSHA or NIOSH method. And so this allows you to, if you don't know anything about how to sample, you can go to this page, click in the chemical that you're trying to sample for, and it will pull up what type of method, what type of OSHA method you'll need to use, what type of NIOSH method that you should use, and it will also tell you what type of media that you should order or use. So it's a pretty handy website to figure out how and what to sample for, and how to go about it. At the end of the day, though, if you're at an institution and you have to do some chemical sampling, your lab that you're going to send the samples off to, they should be your best point of contact. All you have to do is call these guys up, tell them what you're sampling for, tell them what you're trying to do, and usually 9 times out of 10 they'll walk you through how to sample, what media. They'll send you all the media that you need, how to calibrate it, how to basically do everything. And that's really the best or the easiest way to do it.
Very good. Any other questions or discussion points for either Will or Alan? Guys, I know we had a question that came up in the chat last week about quenching some of the cyanide salts. Did you guys have any chance to do a little research on that one?
So I briefly spoke with Will on this, and I think we might need a little more information about what this specific reaction is. But I think the consensus is that if you have a reaction by quenching it, you're essentially stopping the reaction and making it more safe for the folks that might be handling that waste stream. So I think in most instances that would be something that would be recommended if it can be part of the process. Unfortunately, I don't have any researchers currently performing that type of work. Do you agree,
Yeah,
Will?
that's right, Alan. The term quenching is sometimes, it's a general term, but at the end of the day, it usually means to stop a reaction after your workup, and after you're trying to isolate your product, you quench the reaction, and usually you'll add a water or an acid or a base to essentially stop that reaction from continuing. And then you further isolate your compounds or ship them off for hazardous waste safely.
Guys, now's your time. You got Alan and Will for another 5 minutes here. Any other questions or discussions about any of your activities or programs related to chemical safety, industrial hygiene, hazardous waste, or environmental programs?
Not to butt in here, Alan, you probably know, well, I know you know this question. That's why I'm asking. Can you remind us at what point you can neutralize a reaction to basically you're in the lab and you neutralize say an acid. Can you then dump it down the sink, or do you ship it off for waste? Can you remind us about that?
Well, that might depend on the institution. I find as a good management technique that I typically would not ask the lab to do their own neutralizations unless they have specific equipment, say a ventilated sink where they can control any fumes or gases that are generated. And they would also need to take into account any heat that might be generated from doing that simple acid-base neutralization. So typically, if one was writing up a procedure or doing the hazardous waste determination for that process, it may be that the EHS group would collect the waste, take that to a more appropriate facility, and then run through and do the neutralization, and then do some type of sampling before that material, or that neutralized material, is discharged to the sanitary sewer. So that's kind of how I like to do it here. I have a couple of different locations where I have nice stainless steel sinks that have the proper engineering control. They have the ventilation, or they're hooded sinks, and we can perform those neutralizations in those type of areas.
Very good, guys. Thank you very much. For everyone, please keep in mind that the week 2 asynchronous content is always available. If you haven't gotten to it yet for any reason, you can still access all of that material going forward. And we will be monitoring the discussion board from week 2. So if you have other questions, concerns, feedback, or discussion items, please add it to the discussion board from week 2. And we'll get those questions or concerns, discussion items over to Will or Alan for any responses that are needed from our side. Guys, thank you very much for your time. Really appreciate it. You're off the hook at this point. And it is my pleasure to transition over to our next section of live virtual presentation on biological safety basics. And I'm going to ask Dr. Kristen King to introduce herself and tell you about herself, but I will start out by saying congratulations to Kristen. Last week was a massive week for us in that we officially withdrew from the federal select agent program. So she's going to tell you more about our program in a few moments, but we, until last week, had an active select agent program that took a lot of care and feeding and support to be safe, secure, and compliant with the federal rules on select agents and toxins oversight. And we had a retiring principal investigator, and so we were able to support her in closing down her laboratory and successfully withdrawing from the program. So kudos to Kristen and her team. Congratulations. And this brings a significant, what we call A-level risk off of our compliance ledger sheet, which makes me happy. Kristen, it's all yours. Take it away. Thank you for your time.
Well, thanks, Scott. Congratulations to you too, because it's a big thing off of your shoulders as well. Let me share my slides real quick. Okay, guys. So my name is Kristen King. I'm the Biological Safety Manager here at UT Health Houston. I've been with our department for about a dozen years, I think, so far. I've kind of bounced around a little bit, but I started in biological safety here mainly as a way to transition my career from outside of the laboratory. So I worked in BSL-3 infectious disease select agent research at another institution and for several years, and kind of was looking to transition out of the lab and moved into biosafety. Had the privilege of coming here and working for biosafety. While I was here, I went and completed my master's and doctorate in public health in environmental occupational safety. My bachelor's degree is in microbiology. I've kind of done a couple different roles here. I've kind of been the bridge between biological safety and clinical safety, having the opportunity to really work on our clinical safety program as we kind of grew in our outpatient clinic practices here within the greater Houston area. So it's a great opportunity, I think, in many ways, as we have learned over the past, several years. Biosafety and infection prevention kind of get shared responsibilities, especially on the academia side. So I am professionally credentialed in both biosafety and infection control, as well as public health and healthcare safety. If you have any questions, please reach out. I'll try to monitor the text. If you come up with them later and want to put it on the chat, I'll be happy to answer any of those things for you guys. So we're going to go ahead and do our biosafety basics. Again, we have an hour and a half to get through quite a bit of content. So we do have some additional items for you on the site that you can look through, and it gives more information and specifics on certain topics. So what we plan to cover today is just a little bit of the history of biosafety and how that led to our current regulations. And then kind of doing a general overview of microbiological hazards and molecular techniques that are associated with a lot of the work that we're doing in regards to overseeing the work that's done at our institutions. We're going to look at the risk groups for agents, and then how do we assign biological safety levels, and then how to do a risk assessment and risk mitigation. So the term biological hazard or biohazard, those are really used interchangeably. That is an agent of known or suspected pathogenicity in man, animal, or plants. So it's a broad spectrum. In our profession, we use biosafety, biological safety, biohazard control, and infection control, kind of all in the interchangeably sometimes, even though sometimes they tend to have different meanings. So really, a biohazardous agent is going to include the microorganisms, toxins, and allergens that might be derived from those organisms or derived from plants or animals. So those all can create a biohazard. So what do we need for an agent to be pathogenic? So that microbiological agent must be present in a virulent state, so kind of in an active state, if you will. They must have a sufficient concentration. So we'll talk a little bit in this later. We have to have enough of the organism to create an illness. We have to have the presence of a host, something that will actually be infected by the agent. We have to have a route of entry, and then we have to have the host be susceptible to the pathogen. So not all agents can infect every type of host, which is kind of good news in some cases, but so that can be very specific. In defining biological safety or biosafety, we really define this as the anticipation, recognition, evaluation, and control of agents known or suspected to be pathogenic in man, animal, or planet. So kind of to break it down, we can address this as we want to make sure that we have safe handling and containment of our biological hazards. So again, the main two principles are containment and risk assessment. So what do those mean? So containment is, we might think of it as just kind of keeping something in a bubble. And we do that in a couple of different ways. So in biosafety, we use the word containment to include our microbiological practices that we're going to use, our safety equipment, and the facility safeguards. And we do this so that we can protect workers, the public, and the overall environment from any of the biohazardous agents that are used in the laboratory setting. We'll go into these in detail a little bit later on. So the second component is risk assessment, and that is pretty broad. We do that in a lot of different ways as you've learned about the different components of environmental health and safety. So a risk assessment for our biosafety isn't all that different from a risk assessment for your chemicals, etc. So just it's the process where we're going to select for us our microbiological practices, our safety equipment, and our facility safeguards. So those three things that make up our containment so that we can prevent any laboratory associated infections. So we can prevent any exposure and illness to our workers, again, the public, and our environment. So just to talk about the history of biosafety and how we came up with these regulations. We can go back a really long time in history where we can see that bioagents were used in war and different aspects. But really, we started doing these studies. Pike and Sulkin are known for these kind of 4 hallmark studies where between 1930 and 1978, so approximately 50 years, they identified about 4,000 laboratory acquired infections, laboratory associated infections, and those resulted in 168 deaths. And through these studies, they were able to kind of list the top 10 most causative agents, where we had an overt exposure or an overt laboratory associated infection. So Brucella has a very low infectious dose, meaning you don't need a lot of organisms to get it. It also can be aerosolized. Same thing for Coxiella, and then Hep B is kind of the most common for bloodborne pathogens. So they found that these were the most causative agents. Now, keeping in mind that there is most likely a lot more laboratory associated infections that go unreported, especially during this time period. So moving forward from that, Harding and Byers continued that work really for the following 20 years. And they found that there was about 1,267 of these overt infections with 22 deaths. The causative agents were very similar, and they found, or what I think is a really important distinction here, is we were about half and half from the dedicated research laboratory setting and those that were occurring in like our clinical diagnostic laboratory settings. Now here in our academic setting, we don't have a hospital that is strictly run by our university. But some institutions have those, and so we have to be very aware of our clinical diagnostic laboratories as well, and that we might have a role as biosafety there in overseeing those and maybe working with the infection prevention groups within those hospitals to make sure that we reduce these laboratory associated infections. So kind of going back to 1979, Pike had some really good conclusions. Basically said the knowledge, the techniques, and the equipment to prevent these infections are available, but we're still having them. So we have the tools necessary to prevent these. We just have to kind of work better at that. And so like at the time in 1979, there was really no single guideline or regulation that said, these are the best techniques, this is the best safety equipment, this is what we recommend you do to have good biosafety. So these publications really led to these establishment of these biosafety guidelines, if you will. So these guidelines were used to promote the safe microbiological practice, that safety equipment in that facility, so our containment. And those were set up with the National Institutes of Health through the guidelines for research involving recombinant and synthetic nucleic acid molecules. So we just shorten everything here. So we just call those the NIH guidelines. And then the Biosafety and Microbiological and Biomedical Laboratories, commonly known as the BMBL. So those are our two main resources for guidance on these safe practices. So the NIH guidelines are overseen by the NIH's Office for Science Policy, They were first released in 1976, and then they have been most recently updated in April of 2019. They really set the code for practicing for the construction and the handling of recombinant DNA molecules, and then organisms or viruses containing our DNA molecules. So if we're manipulating any of the organisms with recombinant DNA, this is the guidelines that we look to. The link is here for those guidelines. Again, on the webpage, we provided an additional supplementary PowerPoint slide to go further into the NIH guidelines. What do they mean? What is required for an institutional biosafety committee, and kind of how the types of studies are classified within the NIH guidelines. We'll talk about that a little bit later, but I'll reference to go look at the supplementary data on that. The BMBL is a joint publication between the CDC and the NIH. It was first released in 1984. And we're now on the 6th edition, which was released in June of 2020. And this really, the evolution of the NIH guidelines really set the foundation for the BMBL. And then, so this sets the codes for the biosafety practices. So for both research laboratories and for clinical diagnostic laboratories as well. We have, as biosafety professionals, we have a professional association, the Association for Biosafety. I'm sorry, the American Biological Safety Association International was founded in 1984. So 1984 was a big year in biosafety. And so this helps professionals grow. They offer the professional certifications. They provide a way to share information with biological safety professionals. And so it's a really great organization. They also have resources that are available. So whether it's publications through the Applied Biosafety or different training resources and safety, biosafety videos. They have a risk group database, which we'll talk about risk groups in a little bit, as well as a laboratory-associated infection database, which is a great reference. We also have local kind of charter organizations. So depending on what your area is, you can look to ABSA and look at our affiliates to see a local or more local resource for you to provide some of these trainings and resources as well. Okay, so let's talk about some microorganisms now. So by size, prions are the smallest. Then we get into viruses. Our bacteria is a little bit bigger. And then we get to a red blood cell. So when we say microorganisms, we're talking very small things. We just want to highlight here that a lot of these items, right, it's not something that we're seeing. It's really, you can't walk into a laboratory setting and say, oh, there's a virus on this counter. We have to be vigilant that people are working with these things on a scale that is not able to be seen by the naked eye. So let's talk a little bit about some of these microorganisms, just giving a broad overview. If you're not familiar with prions, they are a proteinaceous infectious particle. They basically are this normal folded protein. They can form this abnormal variant where basically the circular protein structure deforms. That can cross the blood-brain barrier. We have some prion diseases in humans. We have Kuru and Creutzfeldt-Jacobs disease. It also forms scrapie in sheep and goats, chronic wasting disease in deer, elk, and then bovine spongiform encephalopathy in cattle. There are many viruses. They are going to contain their fundamental genetic information is either DNA or RNA. Uniquely, they have to utilize a living things cells and machinery to replicate themselves. They can have a variety of structures. They can have, so typically we have these kind of three parts of the structure: a lipid envelope that they receive from the host cell. Not all viruses have this. They have a capsid and then a core which contains their genetic material. Bacteria are single-celled prokaryotes. They have no membrane-bound nucleus or organelles. They have circular DNA called plasmids, which help transmit various traits, usually associated with antibiotic resistance. They are diverse, just like viruses are. They come in a variety of shapes. What you see pictured here is a bacilla, like a rod-shaped. They come in little circles and spirals. If they have a cell wall, it can be made of peptide glycan. They might have a flagella for movement. They might have fibrae, which just helps them attach to other cells or each other. And then they might have a pili, which helps transfer that DNA between bacteria. So they can range within the scope of bacteria. Protozoa are unicellular eukaryotic organisms. They have an ecological importance in that they are very much, they get their source of food from breaking down other things. So this is kind of a biomass control and involved with decomposition. They usually live in water or soil. Some can be pathogenic. And they can reproduce either asexually or sexually. Fungi are eukaryotic. They have a chitinous cell wall. They can be single-celled or multicellular, and they usually grow as these filaments. They are going to be living on or in other food. So they can also be parasitic in some instances where they're basically drawing up all the nutrients from their host. Okay, let's talk a little bit about toxins. These are poisonous byproducts of a microorganism, a plant, or an animal. So they can be produced naturally in animals by algae, by fungi, by bacteria. These can be part of a virulence factor for some bacteria. We can have endotoxins, which are derived from LPS cell walls of bacteria. So this it's just part of the bacteria that they naturally secrete these endotoxins. And then we have exotoxins, which are secreted from the living organisms or released after cell death. So part of the kind of how the bacteria works is that these toxins are released while they kill the cells that they're infecting. Some of these specifically, botulism neurotoxin. It is a spore former. Types A, B, E, and F are most implicated in human disease. It can cause life-threatening paralysis. And the major risks here are with aerosols and needle sticks, although it can be absorbed through non-intact skin. There's usually therapy available, and laboratory exposures have been observed in this, specifically when they've done necropsy on animals that have been exposed within 24 hours to an aerosolized toxin, and then they're getting aerosols from the fur as they do the necropsies. Infratoxins from Staphylococcus aureus are usually important with food poisoning or toxic shock syndrome. They have also been noted in the laboratory through ingestion, needle sticks, inhalation, or that mucous membrane contact. It might be present in clinical samples, as well as the fluids and tissues of exposed animals. Shiga toxins have can be found from a Shigella and some E. coli. So most of these are from ingestion, so severe abdominal pain and watery diarrhea. So it's just another component that we need to be aware of when we're working with these organisms. Another component that we have to think about is, can all of these organisms be decontaminated the same way? Do any of them have special resistance to any of our decontamination efforts? So the answer is yes. So prions are the most resistant. They can have been shown to be inactivated with autoclave, but it's usually quite a bit higher temperature and longer runs. You also just can use a very much stronger chemical exposure. Bacterial spores are next kind of in the resistance. We tend to need to have a sporicidal agent, chemical agent. And then we have Coccidia mycobacterium, which are a little bit more resistant, and we typically have to have again chemicals that are shown to be shown to be effective against those specific organisms. Here we can note that kind of the differences in some of the viruses. So our non-envelope viruses, such as norovirus, are a little bit more resistant to decontamination than our envelope viruses like HIV and SARS-CoV-2. And even those non-envelope viruses are a little bit more resistant than fungi and our vegetative bacteria. So it's important when we are establishing our practices for biological agents that we keep this in mind. So let's step away for a second and just talk for a minute of another component that might be part of the research done at your institution is work with stem cell research. So this is a very kind of active part of research because this can be used for identifying any potential therapeutics, being actively used as treatment for a variety of diseases, as well as being used to have cell differentiation to then start looking at various birth defects. So this can be part of your research that you were reviewing in terms of biological agents. Now, a lot of what our research is doing is not just looking at an infectious agent, but looking about how that is affecting human cells within a live model, such as an animal model, and really looking and trying to get down to the components of what is happening, right? So not only are we looking for items that we can visibly see, say an animal is surviving an infection, we want to get down to what is causing certain reactions to happen on the cellular level. And a lot of how they study this is through molecular biology. So we need to kind of understand the paths that they're going to use during their research to understand if there's any hazards associated with this molecular research. So essentially molecular research, what we're saying is they're going to take DNA from the cells and they're going to change that to RNA. That's called transcription. And then from RNA to protein is translation. And the protein is the work, is performing the work of the cell. So that's the action that we want to evaluate what that protein is doing to cause a certain outcome in the cell or the animal, etc. So a lot of what we're going to do in regards to molecular biology is take that DNA, we're going to sequence it. We may amplify it with polymerase chain reaction. We're going to separate it with electrophoresis and chromatography. We're going to also attach it with chromatography. We might modify it, so we're going to alter it in some way. And we can also use it to search with. So through blotting and screening and probing, we can look and see if our sample has certain genes that are present. through this process. So essentially, proteins, again, perform that primary functions within the cell. So they're going to have growth factors. They're going to tell you what structure they need. They're going to be involved in signaling and transport and cell death. So they can tell you a lot of what's going on with the cell based on what their reactions are. So again, we can identify those proteins to find out what their function is. We can purify them and modify them, characterize them. We can look at all the components of them. We can formulate new proteins and inject them to see what the ultimate outcome would be from that, so we can determine the function of that protein. A lot of our experimental processes kind of look like this, right? So we have a bunch of our reagents. So that includes our plasmids that are going to have our DNA, our tissue or cell culture. We're going to have it, all of the growth conditions for those cells. So all of our media with all of the nutrients that it needs. We're going to grow our cells and infect our cells or our animals. And then we're going to harvest them. We're going to do some kind of chemical extraction with physical disruption, and then be able to fix either those cells, get the outcome protein, and analyze those through various techniques to determine what the result was of changing that protein function. So in that way, we really need to kind of know the process that they're going through for the molecular biology, so that we can determine if any of those hazards are associated and what doing that with an infectious agent might cause. So we're going to move on now to a little bit of our risks. So basically, risk occurs when people interact with those hazards, and our function, either probability of that adverse event occurring, and then the consequences or the severity of that event, right? We kind of recategorize this a little bit as knowing what the hazardous agent is, what that agent hazards are, what we're doing with those, what activities we're going to perform with those, and then what is the likelihood of exposure, and what is that probability, the severity of infection. And we're going to use all of those to determine what our containment is going to be. There's a variety of methodologies that you can use from the BMBL and the NIH guidelines, as well as some other resources. And so really our goal is to provide that safe practices and the facility requirements for that very specific protocol. So what we want to consider is the agent, how severe it is, what those procedures, and then the experience of the person performing those experiments. Some other factors to consider is pathogenicity and virulence, the route of exposure, that concentration that they're using compared to the infectious dose of the agent, the stability of that agent in the environment, and then treatment options, and if a vaccine is available. So normally when we talk about host susceptibility, we assume that we have a normal host. But we can have some changes in that depending on age and gender and vaccine status. There might be some reproductive risks. And then we have to remember that we can have susceptibility between animals and plant pathogens as well. So how do we determine what the risk group is for our agent? So there are a couple different databases that can be used. There's the NIH guidelines. These resources here will all tell you what the risk group is for your agent. So that's going to describe the hazard posed by that infectious agent or toxin in the laboratory. So risk group 1 is our lowest risk. So no disease associated with humans that are not immune compromised. So really, there's really low risk of infection, right? So these are common lab strains of E. coli or yeast or adeno-associated viral vectors. Risk group 2 goes up. So now we have where we can potentially have an infection in humans, but we call that controlled. Treatment is readily available. Disease severity is pretty low, and the risk to the community is low. Usually these are pathogens where we have a really high infectious dose, so we need a whole lot of it to cause infection in humans. These are generally our kind of GI type bacteria, bloodborne pathogens. Risk group 3, we've now gone up in severity. So now we're causing a moderate to severe disease in the individual, and we have an increased community risk as well. These also tend to be associated with lower infectious doses, so we need less of the organism to get sick, and we have an inhalation hazard associated with these now. So SARS-CoV-2, tuberculosis is where these tend to fall. Risk group 4 is our most severe. These usually are lethal outcomes. There's generally not treatment available, very high mortality risk, very high individual risk, and then the perception of community risk increases as well. So Ebola, Marburg. So what do we want to consider when we're talking about the actual procedures that they're going to do with the agent, right? Where they're working, the scale. Is it really low volume or really high volume? Aerosol potential. So when we're talking about infectious agents that are associated with liquid, when we're doing any of these items, we're pipetting, we're vortexing, we're centrifuging, we're shaking, sonicating, that potentially could cause an aerosol of our agent. It can cause bigger droplets, but also smaller droplets. We want to be considerate of our use of sharps and our PPE, our containment equipment. And I failed you guys a little bit because I didn't provide a lot of information on biosafety cabinets in your supplementary, but I would encourage you to look in the BMBL. There's a whole section on biosafety cabinets. They essentially protect you by directional airflow. And they're a little bit different from a chemical fume hood and from, say, a laminar cabinet that would be used for, say, pharmaceuticals. So if you're not familiar with biosafety cabinets, the BMBL has a great resource. We also want to be considerate in these manipulations if we're doing recombinant and genetic manipulation. As we talked about, we can be inserting genes, but we also want to be reviewing what are we transferring any antimicrobial resistance? Are we now, as our gene of interest an oncogene, can it potentially be triggered to cause cancer? Is it going to produce a toxin? Could we potentially be increasing our virulence or extending host range? This is some of the information from the NIH guidelines. These are the different sections that we classify the recombinant work as. They have a lot more information in the guidelines, but generally we tend to fall within the 3D and 3E range when we are talking about academic research that is not involved with, we do a little bit of human gene transfer, and some academia do a lot more, but typically in the lab setting, we're in 3D and 3E. So just want to note with biological toxins, those depend on really their activity. They are mentioned in the NIH guidelines and then within the select agent regulated toxins. So let's talk about select agents just for a second. Again, you have supplemental information that provides a lot more information. There is a specific list of agents that are classified as select agents and toxins. They are considered of concern for use as weapons of mass destruction or dual use of concern. There are some strains that are exempt, and with the toxins, it's very volume specific. So that is something to be very aware of if you're dealing with any toxins. Dual use research of concern is essentially any research that could be potentially used for nefarious purposes or misused for nefarious purposes. There is a policy from the United States government. There are some additional policies that are being in discussion now. So I would be saying if this is your area, expertise to be aware of some of the potential state and federal regulations that may be coming in use to dual use research of concern. And so experience, when we're talking about how that plays into our risk assessment. So each person might need to provide some personal proficiency, what their past experience was. Is this a new technique? Are they skilled in it? Do we need specific training on that technique from someone that's created it? Do they need animal training? All of those things. So now we're going to talk, how do we define our biosafety level? And just remember, our biosafety level does not always equal our risk group. Most of the times they are pretty much in line, but depending on what the procedures may be, they might fluctuate. If you haven't seen this before, this is kind of a basic hierarchies of controls as a way to create controls for any kind of hazard. But in terms of biological, a lot of times we can't eliminate the hazard because that is what we want to study. Sometimes we can substitute because we can use a strain that is non-pathogenic that still presents kind of the same processes as far as infection. Engineered controls is kind of going to be a big one for us. This is a way, what equipment can we use to isolate the hazard and the person? Our administrative controls, how we change how they do work, big one for biosafety. And then PPE. So we'll talk about these in terms of containment. So laboratory practices and technique, our safety equipment, and then our facility design. We call our safety equipment and our PPE our primary barriers. So our primary barriers being our biosafety cabinets, also our safety centrifuges, and then our facility, like think of the outer bubble, lab doors and windows and ventilation. That's what we consider that. So our primary, again, as our biosafety cabinets, using that directional airflow to keep the hazard and the aerosol generated from the work separate from the worker. Safety centrifuge cups or rotors allow us to be able to centrifuge liquids that contain infectious material and have those be sealed and only opened in the biosafety cabinet, so that if there's a break or a leak and there's any aerosol generated, we still have that in containment when we open those. And we'll talk about PPE in regards to our different biosafety levels. When we talk about secondary, it's usually as their separation from the lab from public access. What about controlled access? Do we need separate buildings? Do we need some kind of decontamination systems for the facility? That ventilation for directional airflow? And do we need HEPA filters for the exhaust for the units? Things like that. So we're going to go through each biosafety level. So our biosafety levels is a practice of all of those containment practices, right? The safe microbiological practices, our equipment, and our facilities. We go BSL-1 through BSL-4. So just like risk assessment, 1 was the lowest. We kind of go 1 through 4, 4 being the highest. Again, these don't always mean the same thing. However, they do align. So a BSL-1 is going to be a space where we can know that the microbes are not normally going to cause disease in humans. So very low potential exposure. And then we go up to BSL-2. We are most likely working with a risk group 2 organism here. And then 3 and 4. So we're going to go through these specifically, and you can kind of see how the different practices increase for each level. So if we start with our BSL-1, this is kind of our basic laboratory. We're going to start with our standard microbiological practice, right? So they still have to be trained, basic safety. They need a safety manual. They may need immunizations based on what they're working with. We need to have signage on the laboratory door. So it may not be that we're dealing with an infectious disease, but we might have chemicals and radiation. What are the other items. This is general kind of HAZCOM that needs to be done. We want to pull back any long hair when we're working. Our PPE is going to be lab coats or gowns, protective eyewear and gloves. And when we use those gloves, we want to make sure that they get changed whenever they're contaminated or torn, that we don't reuse them. And then we take them off in a manner, as you see in the top right, where we're not contaminating ourselves as we take off our PPE. We want to wash our hands before and after we leave the lab. There's no eating, drinking, smoking, applying cosmetics, handling contact lenses, or storing or eating any food or drinks in the laboratory. There's no mouth pipetting of any kind. We have a sharp safety policy, so we're properly handling and disposing of our sharps. We still want to minimize the creation of any splashes or aerosols, and we want to decontaminate our work surface after we complete work, if there's any spills or splashes, and if there's any waste generated. Now, I would always decontaminate my work surface before I start, so that I make sure I start with a good clean surface. That's a good practice as well. We also want to have a pest control management program, and then we don't want to have any additional animals or plants that aren't associated with the research in the laboratory space. We don't want anything to go from our research project to non-research and vice versa. Understanding that some of the space is divided between like kind of personal computer space and lab space, but we can't have that. We don't want that crossover to occur. Generally, a BSL-1 laboratory will not have a biosafety cabinet. They might have a chemical fume hood and the standard laboratory type of equipment. They would wear lab coats, gowns, protective eyewear, and gloves. As far as the facility, they would have doors just to kind of control access into their space, a sink for hand washing, an eye wash station. The area should be designed to be easily cleaned. So we should have bench tops that are impervious. We should have chairs that are non-porous and that can be basically we want the whole laboratory to be easily decontaminated or cleaned. Any windows to the exterior should have screens at minimum, and they should have adequate lighting. Now, we're going to go up to BSL-2. So we're going to take everything from BSL-1 and roll that into BSL-2 and then add on to it. And this is how we do from 2 to 3 and 3 to 4. So now we have to have a biohazard symbol on the entrance to the laboratory that needs to have the supervisor's name and telephone number, any required procedures for entry, like putting on your PPE before you go into the space. This is so that if anything happens, we can be aware or notify who's responsible, ask any questions if we need to. Access is restricted to the personnel that are trained to handle those pathogens. Workers must show that they're proficient in these techniques. Generally, this is handled through the PI or through the main researcher. We want to have durable leak-proof containers for collection, handling, processing, storage, transfer of any kind of the agents or the materials that would be handled at BSL-2. We want to have routine decontamination of our equipment and our work surfaces, again, after spills and splashes, prior to any repairs, maintenance, or removal of that equipment from that laboratory. Biosafety cabinets are going to be used wherever possible. Say that sometimes we are using a very large piece of equipment, and so it can't fit within the biosafety cabinet. It's not feasible for that work to be done inside the biosafety cabinet. So we come up with alternatives like more PPE so that they can do this work safely. Spills and accidents should be reported immediately to the laboratory supervisors as well as biosafety. And then they should be involved with the medical surveillance program through occupational health so that any immunizations are offered, potentially any baseline serum stored as felt necessary through the risk assessment. So what the equipment looks like, so we have those sealed centrifuge cups or sealed rotors so that we're not opening any centrifuge items just in on the bench top. We're doing that within the biosafety cabinet. That biosafety cabinet, you can see pictured here where this gentleman is standing. These should be installed so that one, because they function as directional airflow. That's how we get protected. So we don't want to have any disturbances of airflow in front of our biosafety cabinet. So when we're installing them, we need to be conscious of room supply and exhaust, as well as walking paths. And I wouldn't place them near a door because that can provide a lot of fluctuation in that airflow that the cabinet uses. These also need to be certified annually. If you're using any vacuum lines, these should be protected with HEPA filters so that the vacuum, whether it's house vacuum or personal vacuum systems, don't get contaminated. You need to have a method for decontaminating laboratory waste available. So for BSL-2s, they don't have to have necessarily an autoclave located within the lab, but somewhere on the floor within the facility. PPE is the same as BSL-1, although there might be additional risk assessments for respiratory protection based on agent, procedures, etc. Now our facility should have doors that are self-closing and have locks. Again, sink for hand washing, eye wash station. Windows now are not recommended to the exterior, and we should have a directional airflow from, say, the hallway into the laboratory. So now we are going on to BSL-3. When we talk about BSL-3, we now kind of call these containment laboratories. So we're going to have quite a few extra practices and facility controls. So now we have restricted access to personnel that are trained to handle these agents. They have to demonstrate proficiency. Any person entering this lab has to be advised of the potential hazards. They have to meet the very specific entry and exit requirements, and they have to be escorted by authorized users. And those have to be for valid reasons, either for scientific reasons or for support purposes. All of these people that are entering the space need to be informed of the signs and symptoms from the organisms that are going to be used, and receive occupational medical services as needed for consultations. You have to have a system for reporting and documenting near misses, accidents, and exposures. This again is immediate reporting to the laboratory supervisors and biosafety. Again, all of our biological materials must be in durable, leak-proof primary and secondary containers. We now all manipulations must be done in the biosafety cabinet. If it can't be done in a biosafety cabinet, we have to think about some other engineering controls and a way to contain these, or we have to severely increase our PPE. We now have to have inactivation procedures that must be documented and verified for any removal from our BSL-3 to a lower BSL-3. Sorry, to a lower BSL level. We have to have a way of decontaminating our entire laboratory in case we have gross contamination. If we now have significant changes to the use, so we go from one agent to another. If we do any major innovations, and then for maintenance shutdowns. So let's talk about our equipment. We still have to use our sealed centrifuge cups or rotors, our biosafety cabinets, those HEPA line, HEPAs for our vacuum lines. This is a picture of that here, if you are not familiar with that. We now have to have a method of decontaminating our laboratory waste within the facility. So we have to have an autoclave within our BSL-3 facility. They can't remove the waste from the BSL-3, take it down the hall in the elevator to the BSL-2 autoclave, okay? Our protective clothing has increased. It's changed a little bit. So instead of a standard laboratory coat that closes in the front, we now have to have a solid front. So this can be in the form of wraparound gowns or gowns that tie in the back, generally a gown of some sort. Reusable clothing has to be decontaminated prior to laundering. So if you have any of these items that are reusable, we have to autoclave those out before we can send them to laundry. We now are requiring 2 pairs of gloves to work. Eye and face protection. Respiratory protection is usually considered at BSL-3 because we are now dealing with agents that have a greater potential for an aerosol inhalation. And we also add in shoe covers to the facility requirements. So what does our facility look like? Again, these are specially engineered and designed laboratories. So the lab is separated from areas that are open to unrestricted traffic flow. So your BSL-1 or your BSL-2 laboratories can be off the main hallway. Here we want to kind of tuck this laboratory away from the normal traffic flow. So it might be down a hallway, within a hallway, kind of a deal. Lab access is restricted. And then you have to enter through 2 consecutive self-closing doors that create this anteroom in between. So in this picture here, you'll have an outer hallway here. You'll enter through this door to an anteroom where you would change into PPE. This door would close, and then you travel through this door into the main laboratory. Our sinks now have to be hands-free, so a foot pedal of some sort. Eye wash stations must be readily available in both spaces, so usually in the anteroom and in the main lab. These are designed for decontamination. So our walls and our ceilings all have a smooth finish. Floors are usually slip resistant, so everything's kind of seamless and sealed. It's we don't want to have any kind of nooks or crevices that things can get stuck in. So we have to make sure that all of the everything's sealed from our lights, light fixtures, anything that has protruded through those walls. Windows must be sealed if there are any, very unlikely. We have to have a ducted air ventilation system. So this is our directional airflow. So we go from clean to contaminated. So essentially from the hallway into the anteroom and then into the lab and ventilate it out. This has to have a visual monitoring device so that you are aware that directional airflow is occurring properly when you enter the laboratory. And then the lab exhaust is not recirculated to the building. So it goes through HEPA filters and then it goes out of the building exhaust. Again, we have to have our biosafety cabinets and our HEPA filters within the biosafety cabinets, as well as for the exhaust systems, have to be certified annually. We actually have to have our BSL-3 facility design and operations be verified and documented prior to the initial operation and then annually thereafter. So this does require an annual shutdown of work, decontamination, and then verification of all of the parts that make up our facility design be verified. BSL-4s kind of come in 2 categories. We can have what we call a cabinet lab, as you see on the left, or a suit lab. So essentially what happens in a cabinet lab is the use of these class 3 biosafety cabinets. Talk to a little bit about how those work. And this allows you to do this work in a little less, it's more restrictive as you're doing the work, but you don't have to wear as much PPE. And then in our suit labs, you are using a positive pressure suit for protection, and the lab work is done kind of at BSL-2 level. So again, as we're talking about our standard microbiological practices, we have, we're going to use all the same ones as we do for BSL-1, BSL-2, BSL-3, and now we have, again, Much of the training and the access restriction for people going in is the same. The training for BSL-4 is many months long to have someone be trained to work in and handle pathogens at that level. Again, we have to have these systems for recording and documenting any near misses, any accidents, or any exposures. Much like BSL-3, we have to have those durable leak-proof primary and secondary containers, and as well as inactivation procedures have to be documented and verified for any removal from BSL-4. And then all waste has to be decontaminated by a verified method prior to removal from the laboratory. Again, much like BSL-3, decontamination of the entire lab has to occur. If there's gross contamination, any significant changes, maintenance shutdowns, renovations annually as required. Some additional ones is there has to be a logbook for entry-exit, and then there has to be a daily inspection of those essential containment and life support systems, like the suits that are being worn. Only necessary supplies go in. These usually are only coming out through a dump tank. I really, both at BSL-3 and BSL-4 is encouraged to only take the essential supplies in and anticipate that those be remained in those facilities. So let's talk a little bit about a Class 3 biosafety cabinet. Unlike a Class 2 where you can just have an open spot to put your arms through to work, you're going to be working through these very thick gloves. Essentially what happens on one end is there is a double pass-through door. So it's kind of like submarine doors. You open one end, they're both gasketed. So you open one end, put supplies in, close that, and then open the other side from the side that has these gloves in it to be able to go through. On the other end, as this picture is indicating, there's a dump tank. So you can remove items from the Class 3 biosafety cabinet, or usually there's an autoclave that is attached to the other end of that. So past this gentleman here would be an autoclave attached to that. The Class 3 glove box has HEPA filters for supply air, as well as 2 filters on the exhaust. And then it goes through building exhaust. These are under negative pressure, and these gloves have to be inspected prior to use, as well as an annual recertification. So as you can see with this gentleman, the PPE is a little bit more relaxed if you're working in a cabinet lab. So solid front gown, dedicated scrubs, Tyvek overalls or coveralls is usually what is used. Shoe covers and gloves. Any prescription glassware that goes in through a BSL-4 has to be decontaminated before you kind of do the exit out shower, which we'll show you. So I just want to kind of, if you haven't seen this before, what kind of happens with a BSL-4, you enter into a locker room area, you have to remove street clothes and put on dedicated scrubs. After there's you have to go into where the suits are being stored. You have to inspect those and test those each time that you go in. You do a pass-through shower and then go in to do the lab work. Again, working much like a BSL-2 in this situation where we're going to still use our biosafety cabinets. When we complete our lab work, we go through that chemical decon shower, remove the suits, we remove the scrubs, and we do exit shower, change back into normal street clothes, and then exit. So it's a lengthy process. Let's see. We do still want everybody to use any manipulations in the biosafety cabinet. Anything that's aerosol generating devices should have a HEPA filter on them. We still use those seals, centrifuge rotors, or cups. And then those positive pressure suits, again, you are wearing dedicated scrubs underneath. You verify each of those suits for operation and performance each time you go in. Have a pair of inner gloves on as well. So what does the facility look like? It's pretty significant. It's usually a separate building altogether. Access is restricted. Again, they have hands-free sinks at all locations, eye washes, much like a BSL-3. Those, think of it as kind of a bubble within. So the walls, floors, and the ceiling are all kind of like a sealed inner shell. All of these penetrations are sealed. There's minimal furniture located within these, and then the windows, if there's any, need to be break resistant and sealed. You also have to think about a communication system from within the laboratory outside. I'm sorry about this picture. I just wanted to illustrate that usually when there is, you can see this second row here is the laboratory. We have 2 whole floors worth of ventilation protection, and then we have usually a couple additional floors that have to deal with generating or inactivating all of the waste that is generated from these laboratories. So pretty significant maintenance dedicated to these laboratories. Everything has to be automatically activated on emergency power. You have to have monitoring systems for the air supply, the exhaust, life support alarms, entry-exit controls, and all of your security. Centralized vacuum systems are discouraged. So if you have to have that suction, like we were talking about for the other laboratories, you have to have 2 inline HEPA filters as well as overflow collection. So you have to do a little bit more in regards to that. The ventilation is non-recirculating. So all of this kind of is clean supply in, exhausted out under negative pressure. There's redundancy in the supply and the exhaust, and these must interlock so that we don't have any failure. And then again, much like BSL-3, these are HEPA filtered prior to exhaust. You have to have a liquid effluent decontamination system, which is kind of what you see in the bottom of that picture. And you have to validate those as well. And then these also have to be verified initially, the design and operation, and then annually as well. Okay, guys, I was afraid we were going to run out of time, but it looks like we have a bit of time. So I'm going to stop for a second and see if there's anything in chat. I can't. Okay. So I'm going to go through a case study with you guys. So feel free to put this in chat, which I can't see in this current slide set. But kind of wanted to give you an idea of how to go about doing a risk assessment. So it was think of this as a researcher coming to you and saying, our primary goal is to use group A Streptococcus for our research, and we want to understand the bacterial and host mechanisms contributing to a wide variety of disease caused by group A strep, including its carrier state. So what they state that they want to do is culture bacteria, test antibiotics. They want to do protein identification and gene sequencing. They also want to downregulate genes suspected of virulence. They want to look at in vitro characterization of infections in human epithelial cells. And then they want to look in vivo infections in studies in mice. So the first thing that we might want to do is look up what our risk group is for Strep A. And then think about these procedures as we determine what is the best biosafety level, which includes all of our containment procedures to be associated with that. So we have looked it up and found that it's a risk group 2 organism. So some of the characteristics is that it's exclusively a human pathogen. It's aerobic, it's non-modal, and it's not spore forming. We usually see transmission through direct contact with body fluids, inoculations, and potentially from infected animal feces and the bedding that might be contaminated. We have an unknown infectious dose and disease severity ranges, although there is a range of effective treatments. So they conclude that it's a low to moderate community risk. There is a lot of known LAIs, especially when we think of aerosol generating procedures. So what we are trying to figure out is, can we use this at BSL-1? Is that the appropriate place for this organism to be used at based on our definitions of containment for BSL-1? Unlikely. We probably put this in BSL-2 because that is telling us we're going to do any manipulations of our stock filters in a biosafety cabinet. We really want to hone in on those aerosol generating procedures to make sure that those are only being done in a biosafety cabinet. Most of their procedures would be growth of that, which is usually going to be in a shaker incubator, which may not fit into our biosafety cabinet. So we may want to recommend that they use filtered stoppers on those shakers to prevent any aerosolization. Let's see, when we're talking about infection with animals, so that is a concern. After those animals are infected, we're talking about some of those can have infected animal feces and bedding that can then be disposed of with our animal care staff. And we want to protect our animal care staff as well. So we don't want to have them treated as a normal cage. We want to be able to make sure that we are aware that there's a hazard there. If they're dumping out bedding without any protection, that could be a potential source of an exposure. So we would most likely put this at ABSL-2 to make sure that our animal care staff is aware of the additional hazards that are associated with that part of the procedure. Again, I would remind you that those additional slide sets for the RDNA guidelines, as well as how to set up your institutional biosafety committee, are available for viral vectors, for disinfection, decontamination, and sterilization, for shipment of infectious substances, and then for select agents and dual use is available as well to you. If you need more information specifically on any of those, as well as the portion of the BMBL that discusses our biosafety cabinets. I'm going to close this out in case, so I can see the chat, and if there are any questions, I'd be happy to take those at this time. So someone asked, what risk group is prions? Prions are risk group 2. We tend to have additional procedures for those because, one, we have additional procedures in regards to decontaminating and handling hazardous waste out of that facility. We have isolated them a little bit more than a standard BSL-2, simply because that has been the request of the community here, and that's what our IBC, and we had the space available. And that has seemed to work out very well. We have different prions that we work with. So we have some of the human prions, which are kind of in that isolated facility, and then the animal prions, which are kind of mixed in with the main lab areas. We have made those procedures and the safety precautions that we put in for those places very visible to our community, so that they know that we are having these extra precautions for those.
There is a second question, Dr. King, that reads, do you have any recommendations for someone that will want additional training for biosafety officer?
Sure. So we do offer a week-long course as a biosafety officer course. I'm sure that the group here or Scott can provide information on when that might be next available. So that's the best resource that I think I can give you at this time. Anybody have any other questions? Any other agents that you guys are concerned about? Well, as we have time, as Scott mentioned at the beginning, we have just withdrawn our select agent registration. Now, just to give you a quick insight into that process, it's not something that you do all the time. Some people may not even have select agent programs, so setting up a select agent program is definitely a challenging aspect just to get used to the processes that are involved. But there are guidance on how to do that. And so we had this researcher, I think she was doing work before the select agent regulations really went into play. And so I was not here then, but we worked through the process to get that registered space. And then it was kind of the status quo, normal procedures for 20 years and maintaining that laboratory. And as you probably are familiar, when we have a retiring PI, a researcher, about a year out, they start to not have lab staff and research maybe slows down a little bit and there's not a lot going on in the space. And then all of a sudden they're like, I'm retiring and bye. And so we have a lot of leftover materials that we kind of are up in the air about. And so there's various processes that we've tried to put in place as far as being aware with our departmental administrative staffs that if you have a PI that's looking to leave, that we have a checklist to do so that we don't have these kind of orphaned materials stuck in our laboratories and we don't quite know what to do with those. For our select agent lab, it's kind of the same thing. We had to, one, determine what stocks needed to be transferred to other select agent registered spaces. And then if not, we had to destroy all the other stocks and show proof of that. We had to kind of remove through autoclaving a lot of the residual materials that were in the laboratory, so pipettes and things of that nature that couldn't kind of be reused. And we spent quite a bit of time doing that. And then doing a whole room decontamination. We do that in-house. And then additionally, we had to do via a vendor a decontamination of our ductwork and our HEPA filters. So again, when you're talking about a PI that's looking to retire that's select agents, we have to consider that they don't really have much funding resource left. And so shipping select agents is very expensive. And some of these closeout materials are expensive, like that decon of that HEPA filter. So who's going to take on the cost of that? How is that planned into the decontamination process? So that was a big lesson that was learned for us here. And let me see, I just have a question. So at our BSL-3, so the question, if you haven't seen it in chat, our BSL HVAC exhaust duct panels required to have appropriate warning labels, even if they're a distance away from outside of the boundaries of the BSL. Yes, so this is what we did. Your ductwork should be labeled at BSL-3, right? So BSL-2, it's not isolated for that laboratory. BSL-2 is, I'm sorry, BSL-3, the requirement is that we have separated ductwork from that laboratory that goes through a double HEPA bank before it's exhausted straight from the building. So all of that ductwork up to those HEPA filters is labeled as biohazard. Hopefully that answered your question. And essentially for BL4, the whole building is would have that labeling. So that is really the tough part. I know that there was a lot of requests during the COVID pandemic as researchers wanted to start working at BSL-3. How do we convert a BSL-2 space to a BSL-3 space? And it's not just practices, it is these actual facility requirements. So going in and, creating doors that are interlocking, creating these negative pressure areas through our, putting in whole new ventilation systems, is something that the researchers may not be aware of, that's the requirement to do that in that space. So we at our institution had to kind of what work can be done in the BSL-3 and inactivated to go down to BSL-2 for, say, molecular analysis. What work that is being done with human samples, how is that classified? I'm sorry, human samples that are COVID positive, what additional items and security measures and safety precautions could we put in place so that we could safely handle this at a lower biosafety level? All of those things can and should be considered for each specific protocol. Okay, question in: if facilities personnel enter a BSL-2 or BSL-3 labs to do maintenance, what are the requirements for them before they enter? One, for BSL-2, there shouldn't be any active work going on. Typically, this is less restrictive for our facilities folks, but they should be aware of the laboratory that they're going into, what those PPE requirements are. Because our BSL-3s are a restricted space, they can't get access to those without somebody that is an authorized user. So what we typically do, if we have to go in when the BSL-3 is an active work, we will discuss the signs and symptoms, what the laboratory is actively working with. Their facilities folks will need to be respiratory fit tested and wear an appropriate N95 respirator as we go in. They have to meet all the entrance and exit PPE donning, doffing requirements to go into that facility to do that work. We do have our annual decommissioning of the lab, where the lab stops doing work and we fully decontaminate it. And we try to work as much as we can during that period for any major renovations, facility type modifications that we need to do. If we can push it off until that time, the lab is fully decontaminated. We have not as strict requirements for PPE. They still have to go in with an authorized person, but the hazard has been eliminated because of the decontamination and not actively doing work. So that's when we change out light bulbs and paint and do all of those things that we need to do. Typically, if there is a major renovation project happening in a BSL-2, trying to think, we've had it's usually like work has been completely stopped in that area. All stocks have been kind of stored away in refrigerators, etc. And really, if you're going in to do any maintenance on any piece of equipment, your biosafety cabinet, having your pipette service, those should be decontaminated before a facilities person or a vendor is using to certify those.
Okay, if we don't have any more questions, we are going to break here. Thank you, Dr. King, for your presentation. And we will be back at 11.
Okay, thank you, everyone. If you have any questions, that you think of later, you can put them in the discussion board and we'll get to those as well. Thanks so much.
Thank you.
Welcome everyone back from your break. It's 10:58, so we'll wait maybe just a couple more minutes for people to come in, but we can start with Robert Plant is here, our next presenter. He will be discussing basic ergonomic assessments. If Mr. Plant, if you'd just like to introduce yourself before we get into the presentation. Thank you.
Okay. Hi, I'm Robert Plant. I'm with the Chemical Safety Program. As part of that, I've done a lot of ergonomic assessments. So I'm just going to go through some basics of ergonomics. All right, it's 11 o'clock, so I'm going to go ahead and get started. I'm going to be talking about ergonomics, specifically office ergonomics, because that's what we see the most of. But the principles are the same kind of across the board. We're going to go over what ergonomics are. I'm going to talk a little bit about ergonomic injuries and why it's so important to have an ergonomics program, identifying some of the challenges, setting up a workstation, and just some general guidance on ergonomic equipment. First, ergonomics is, in one kind of sentence, it's fitting the task to the user. So you don't want to force people to kind of bend over backwards to do whatever they need to do for their job. You want to arrange it so they can do it more naturally. So the average person sits in about 14 hours a day. That's mostly associated with work. And people perform up to 200,000 keyboard strokes daily. That's a lot of movement. And when you have the kind of repetitive motion, it could lead to repetitive motion injuries, which is the most common workplace injury. Yeah, so lack of physical activity is kind of one of the biggest issues in the workplace. Injuries involve things such as swelling, discomfort, insomnia, numbness. It's kind of across the board. It could be anything from soft tissue injuries, like you pull a arm, to things more like you hurt your nerves for compressing your carpal tunnel. And these all don't happen overnight. They happen over years of exposure, which is one of the reasons they're so hard to identify, because it's just kind of creeps up on people. Musculoskeletal disorders count for 29% of all workplace injuries and illnesses that actually require time away from work. This is from old data, but it's only gotten higher. And it's increasing regularly. The average number of missed days of work in 2011 was 16 days per incident. So if these happen regularly, they're missing a significant amount of work. The average lifetime cost of carpal tunnel syndrome, for instance, is estimated to be $30,000. So if you could find ways to prevent these injuries, then you're just going to save money as well as make that person's life significantly better. Catching them early and preventing them is kind of the key. Once it starts, it's kind of going to become expensive to fix. And just kind of knowing what to look for. The most common one is carpal tunnel and cubital tunnel syndrome. Carpal tunnel syndrome is when the thumb, the pointer finger, and the middle finger kind of are numb. That's kind of the first sign. Cubital tunnel syndrome are the pinky and the ring finger kind of tingle and get numb. That's kind of your key thing to look for. The carpal tunnel is in the wrist, so it's usually caused by people compressing their wrist, where the cubital tunnel is in the elbow. So when you rest your elbow on your desk or on your armrest, you're compressing it, and that can lead to the same issues. Other ones, such as tennis elbow and tendonitis from moving over and over. Also, things like herniated disc and sciatica from sitting too much or sitting in bad posture. As workers age, they get better at their job, but their bodies also become more susceptible to injury. Also, people are gaining weight as the years go on, which also makes a lot of health problems. All of these things kind of come together to make very expensive ergonomic related injuries. For example, back injury, the number one reason for seeing a doctor other than a common cold in the US, and this costs the US around $50 billion a year. And just even any injury that's musculoskeletal is just going to be expensive. It's going to require surgery and recovery. and it's going to take people off work for a long time. And all these are kind of caused by repetitive motion. So in a lab, pipetting, people have to do the same thing over and over all day long, pressing keys, typing. Most people do the same job over and over and over for their entire 8-hour work period, and combine that over the life of their job, they're getting a lot of strain. Even if it seems small, like typing on a keyboard, over the years it'll build up. So how could you prevent these? Just general guidelines before I get into actual workstation adjustments. Avoid bending and stooping. You want to have everything in the middle of the body, so it's kind of your power zone. You want the body and location allows for a comfortable neutral position. You don't want people extending. You want people kind of comfortable as they're working. And everything needs to be in easy reach. You don't want people reaching across their desk for their phone if they could put it right next to them. If they're moving something heavy, it's important to push, not pull, and to let the device do the work. If it's on a cart or a dolly, you don't want to, it should be easy to move. And if it's not, then maybe it's on there wrong. And if it can't be pushed, then you may want to look for additional help, like a motorized pallet truck or something along that line. Proper posture, moving your work task, pacing, all these are important to avoid getting injured in the first place. Like I said, if you're doing the same job over and over again, you're eventually going to cause a repetitive stress injury. But if you could break it up, so every 2 hours you can do a different job, that gives your body a chance to take a break. If you're lifting something, don't assume you know how heavy it is. You want to test the weight of the object before you start. And make sure that you know the path you're going is clean. And if you think you're starting to get an injury, make sure you get help. Now, I'm going to go over some things for the assessment, especially exercises and how to adjust a workstation. First thing, when you go and look at somebody, you want to see how their posture. Are they sitting straight? Are they bent over? Is it awkward? Do they have to reach for something? See if they're using a lot of force for something. Do they have to push something or pull something? Is their keyboard kind of not a good one and it's causing them to have to type harder? And also how long they're doing something for. Before you even start the ergonomic assessment, I recommend watching them for 5 minutes or so. It seems like a long time, but if you're watching somebody work for 30 seconds, they're going to look like they're doing fine, but they haven't gotten to the natural habits yet. They're going to be conscious of you watching them. Look for things that are called contact stress, so things that are applying direct pressure, such as maybe they're resting their hands on the desk. Environmental issues. Is the lighting okay? Is it too loud in the office? Is it really cold or really hot? And other psychosocial issues, such as if they're doing a repetitive task over and over again, are they going to be more likely to get injured because they're not paying attention to other things? So when you start the ergonomic assessment, you want to look around the workplace and figure out what is going to be the highest risk. We have checklists that help with that, and just see if there's anything that's going to be causing more issues than other things, and you could start with that. It helps to give the person you're going to be performing the assessment to a self-assessment first. They could look through it and figure out what they think the risks are, because they work in this workstation 8 hours a day, you don't. So they're going to know the area. And then you could find solutions without even starting the ergonomic assessment by just changing some minor things. For the actual self-assessment, this is the form we use. And I always start with a chair, then go into the keyboard and mouse and the monitor, and then just a general environment and basic work practices. Personal information on the top, trying to have this for your own records. This is where I focus on, and then at the end is required some recommended actions. I tend to not recommend ergonomic products when I can avoid it, because ideally you want to teach the person how to use what's already there. Also, on the back of our form, we have a general guideline for the person if they want to do a self-assessment. This kind of shows somebody how they're supposed to set their own workstation. It's good to give it to them first so that you're not completely changing things out of the blue. They have an idea of what you're going to do. The checklist allows for kind of the key areas to be assessed, so we know when looking through it that we have the seat height going to be checked, the back, the depth, the pan tilt, and the armrest position. And you know kind of how each of these, what you're going to look at. And you can mark acceptable and needs improvement. Most of these things can be fixed during the assessment, but some of these may require additional work. Sometimes monitors need extra tools to adjust. Also, I don't like adjusting computers and monitors myself, because when I touch them, they tend to break, and then you've ruined somebody's work. So I usually let them make their own adjustments. Also, make sure all their work saves before you get started, because you are going to be watching them use the computer, and you don't want them to lose any work because as you move the monitor, you touch the power cord. These are some of the regular recommendations we do make for items. The one of these I use the most is the footrest, which I'll get into later. But you want to find items that work best for that person, things they don't have to kind of extend to use, things that make it more usual to use. So for example, if somebody's using the telephone and they have to tilt their head sideways to hold it onto their shoulder, but they need to type while they talk, then you may want to look at giving them a telephone headset. If somebody's looking down to transcribe paperwork on the computer, then a reference document holder will let them look straight. They can just turn their head slightly as they're typing. So ergonomic factors, it may not seem like it's an ergonomic issue, but lighting definitely is. Most lightings were designed for paperwork, not computers. So many offices are way too bright. Ideally, you want to lower the lighting level in the office and then have task lighting for typing, for writing. So when somebody does need paperwork, you turn on their lamp on their desk to give them more light, but otherwise you want to decrease it so that the glare from the computer is not causing eye issue and eye strain. And also you want to reposition any task lighting that is there so it's not directly shining on the monitor, because you want it just shining on where they're typing. Also, this is also energy saving. Lighting is expensive. It's almost half the energy for your average commercial building. And if you could find ways to lower it, then it's going to help the person's eyes as well as help the building's budget. Ideally, if you could have different sources of lighting so that you can adjust how much you need depending on the user's needs. And if you can't get rid of light, then think about installing glare filters or ways to shift the monitor so it's not directly under the light. But that's kind of your last line of defense. Ideally, you want to lower the lighting first. Also, make sure you know where it's positioned. If you have light, like a lamp directly behind the monitor, it could cause contrast issues. If your monitor is behind a window, you have the same thing. So you may want to consider blinds. You want vertical blinds if it's east-west facing, or horizontal blinds if it's north-south facing, just to make it best to get rid of the sun glare so it's not causing people issues. Ideally, you don't want your desk behind a window at all, but if you have to, these are some ways to get around that. Noise is a big one. Sometimes they'll have to work around machinery, and sometimes people just have a loud office, people are talking or so. Headphones are great for this. OSHA recommends 85 decibels for the 8-hour, so you want to keep stuff lower than that. And if it is higher than that, then they have to have some form of protection, such as earplugs or that, and that's something that can be provided. Indoor air quality. A lot of people complain their office is too hot or too cold, and these are just some general guidelines. You want the temperature between 68 to 79 degrees, depending on kind of what the temperature is outside. In the winter, you want it cooler. In the summer, you want it a little warmer. Humidity between 30 and 65%. All of these things, there are equipment to go over, which I think Will went over this morning. We like to recommend people do the 20-20-20 rule. So every 20 minutes, take 20 seconds to look at something 20 feet away. And that just gives people a way to give them eye break from the monitor, so it's less eye strain. Also, alternate the task, preferably not on the computer. So if somebody is typing all day, then take 20 minutes to go do something else, like print stuff or make copies if that's something they could do. Stretches are great. I'm going to go over stretches for a while. This is about halfway through the presentation, so y'all can do these stretches with me to give y'all a break. First, This is what we give people, and this are some basic stretches, just kind of how to move it. I usually don't make them do all these stretches. I just kind of say, find what has the most strain and work on that first. And then if you have time, do additional stretches. But are these going to move? Okay. So first, the shoulder stretch. You put your arm across your body and you pull it back and forth and you hold it for about 30 seconds. This kind of stretches your shoulders and your upper arms. Next is an upper arm stretch. This one you want to sit down. You want to put one arm behind your head and you want to kind of push it down and kind of tilt to the side. And you do this on each side. Chest stretches, put both hands behind the head and bend your arm backs and kind of push your chest forward to kind of stretch that out. All of these should be held for about 15 to 30 seconds and just do them a couple times. Chin tucks. This is a good back of your neck stretch. Put your chin to your chest, hold it 15 to 30 seconds and look up. And you'll feel the tension in your neck. Head turn, another easy one. Just turn your head left and right, but hold it 15 to 30 seconds on each side. And then do it side to side. Kind of want to put your ear to your shoulder. Lower back stretches, sit on a chair. Always want to do these sitting and put your knee up as high as you can get it. Hold it for 30 seconds and put it back down. Standing side stretch, just kind of grab your knee and do it. I recommend doing this near a desk or a wall so you can hold on. Otherwise, people tend to lose their balance and fall down, or maybe that's just me. Wrist stretches, put your hand directly in front of you, put your hand down and pull it towards you. You can also do the reverse where you put it face up and pull it towards you. And you want to hold it for around 30 seconds again. And finally, this is another good one for eye strain. Rub your hands together so they get a little warm, put them up to your eyes, and then kind of push in gently and leave it there for a little bit. Okay, so now ergonomic equipment and workstations. Kind of, if they're bad workstations, then it could cause eye strain from glare, back pain from bad posture, and neck and shoulder pain just from improper usage of your equipment. And how you're adjusting it will depend on the user and if the user has anything else. Some of the biggest factors that lead to ergonomic injury are bad posture, or I should say awkward posture, improper workstation setup, repetitive activities, high force activities, direct pressure, bad physical condition, and doing activities improperly. So you want to adjust the workstation to the user. Some of these are hard because you can't usually adjust a desk, for instance, but you do your best you can with a chair. The first thing you want to do is you want to make sure you see where their elbows and knees are. You want them both your elbows and your knee at about a 90-degree angle. If it's a little higher or a little lower, that's fine. I usually look for between 85 to 100 degrees, just kind of in that range. The average desk is designed for somebody who I think is 6'2", and most people aren't 6'2", so you'll need to do some adjustments. This is where a footrest comes in handy. Even if somebody is tall enough to not need a footrest, it's still a good idea, but it gives them something to push off against. You want the head in line with the spine while their back is fully rested on the chair. You want the chair at a good height, so both, like I said, the elbows and knees are at 90-degree angle. And you want the mouse and keyboard on the same level. This usually isn't an issue, but sometimes if you have a keyboard tray, they'll have the mouse on the desk and the keyboard on the tray, and that's not going to be good in the long run. And you want everything directly in front of you. So you want the mouse next to the keyboard. You want the feet flat on the floor or on a footrest if that's possible. You want the thighs parallel to the floor, so you want them sitting, flat. You don't want their legs under them, kind of propping their thighs up. You want their forearm also parallel to the floor. The armrest is kind of a controversial topic. In older ergonomic recommendations, the armrest was always recommended, and now people are moving away from it because it can compress the cubital tunnel. If you can use the armrest with it only supporting the meat of your forearm, then it's good to use, but most armrests are not designed to be used that way, so it may be best to lower the armrest so they're not tempted to use it, or remove the armrest if they're not tempted to use it. But if they can use it, you do want to recommend it because it does take off shoulder pressure. The backrest, you want it to support the lower back, and you do want them to use the backrest. If they're sitting too far out, they're not getting any extra support. And you don't want them looking up or down. You want them looking straight ahead. The neck should be straight. And their shoulders should be relaxed, not put up near their ears. You want the screen about an arm's length away, so they should be able to reach out and just barely touch the screen. Any closer or any farther, you can have eye strain issues. And this is probably the #1 change I make for office ergonomics. The top of the screen should be at eyebrow level. Almost everybody has their screen way too high. They have the middle of the screen kind of centered with their eyes. But you don't want them looking up and down for the screen. You want the top of the screen at their eyebrows, so they just kind of gently look down with their eyes, not their neck, to see the whole screen. This does not apply if somebody has bifocals. If somebody has bifocals, you want to adjust the screen lower so that they could look lower in the glasses to see it more clearly. And you want a position to minimize glare and reflection, and glare protectors can help a lot with that. Documents that they're typed that they're transcribing from should be within easy reach, and it should be perpendicular to the desk. So you want it up, so not looking down. You want them to look straight ahead and be able to see it. Most monitors aren't this chunky, so it should be easier. The desk, if it is adjustable, you want it about 25 to 29 inches high, depending on how tall the user is. So some basic adjustments. First thing you want to do when you're adjusting it is to adjust the height of the chair so that their knees are 90-degree angle. Then you want to adjust the pan depth. You want about 2 fingers between the back of their knee and the front of the chair. Not all chairs have this adjustment, but it's kind of an important one. Otherwise, their knees could get compressed. Then you want to adjust the backrest height if it is adjustable. Not all are. And you want to adjust the tension. This is another big one. A lot of people have their chair set so they cannot lean back. You actually want to be able to lean back, but you want to turn the tension of the chair. Most chairs have a knob somewhere on the bottom. You want to turn that as high as it'll go, and you could just slightly lean back. So you want the chair to support all of the person's weight and only move a little bit. And then you want to adjust the height of the keyboard if it ends with a negative slope. You want a negative slope because you don't want, you want your arms facing down, not up, and I'll get that into a minute. You want everything within easy reach. So if you look at this little diagram, the less you use something, the farther you're allowed to have it, basically. You want things as close as possible to use often so you're not reaching. If somebody is reaching regularly, you really want to move it. The biggest culprits for these two things are desktop scanners and desktop phones. Because scanners are so clunky, people have them far away from their computer because there's no room, but they tend to reach a lot instead of moving their chair. And that causes a lot of reaching. You want to maintain comfort is the important thing here. You want the arms kind of hanging from the shoulder at a 90-degree angle. You want the head in the straight line, elbows close to the body, not kind of winking out to the side. And you want the feet on the floor or on the footrest. Risk considerations. Most keyboards have those keyboard tabs on the bottom. Those were actually developed when keyboards first came out to help people transition from typewriters to keyboards. In the 90s, they actually took them away because they're bad for your wrist. People complained so much they put them on. Ideally, you actually want to type at a negative degree angle. So you want your fingers lower than your wrist. That's why keyboard trays are so good. But if that's not an option, have the keyboard flat so your wrists aren't up, because that's just going to force your your carpal tunnel into whatever hard surface you're resting your hands on. Also, a quick note on that too, before I get to sit to stand, is a lot of people like the silicone wrist rest. I don't recommend those. Those compress your carpal tunnel. You want a beanbag wrist rest, so it kind of shapes around your wrist. Sit versus stand. Both of these, if you stand too long or sit too long, are bad. You can develop MSDs from standing too long, discomfort, and you get circulatory problems. Sitting, you cause increased cardiovascular disease, obesity, and type 2 diabetes, and increased blood clots. They both have their negatives, but that's why you want to mix it up. Ideally, you want to stand for 2.5 hours of the day, basically, and you want to increase energy expense. That'll increase the amount, the calories they burn, and it's healthier for them because it kind of moves them up and down. The rule, when people get new sit-stand stations, especially if whole offices get it, they tend to treat it as a competition, see who could stand the most. I like to tell people to sit for 40 minutes of the every hour, stand for about 15 minutes, and then 5 minutes of stretching. So you just want them to basically move back and forth. It's also just even adjusting their posture is a good way to give them a break from the computer. Because when they're sitting and standing, or when they're changing their posture, A, it reminds them to have good posture, and B, it gives them a chance to look away from the computer. Encourage frequent micro breaks, just because that's going to be the best way to prevent eye strain and body strain. Footrest, I recommend a lot just because it reminds people to have good posture. And use the biggest muscle group for different tasks. So if you can use your arm versus your fingers, do that. And all of these things don't do any good if the person doesn't do it. So it's good to send them a checklist of what to look for so they can remember the things you told them. Ergonomic equipment. A lot of things are called ergonomic, but very few of them are. There is no governing body for ergonomics. So just use your best judgment when you look at it. I haven't found a good one-size-fits-all. So every time you go talk to somebody who needs something, work with them to see what works best for their body. And you just want to have something that can make these adjustments ideally. If you cannot find a chair that makes all of the adjustments, the most important ones for me is the span depth, the adjustable back support, and the height. Here are some ergonomic resources. Most of this is centered around office ergonomics, but there are plenty of other ergonomic, like warehouse ergonomics or lab ergonomics. All of these follow the same general principles. It's just a different thing you're adjusting. Anybody have any questions?
Thank you very much, everyone. So I wanted to start by telling you a little bit about myself. And then also I wanted to do a quick demonstration with an atomic fireball. So actually I'll hold it over here because of my plant. Anyway, a little bit about myself is I'm a radiation safety manager here at UT Health Houston. And I've been at UT Health Houston for over 25 years. I originally was a Texas A&M Aggie. Whoop, for any Aggies that are on here. But anyway, I got my bachelor's in radiological health engineering and my master's in industrial hygiene, master's of science at UT School of Public Health, and then a doctorate at UT School of Public Health in environmental and occupational health sciences. And so I've been doing radiation safety here for quite some time. And we usually in the EH&S Academy course, we get people that some of them have a lot of radiation safety experience, and then some of them have little to no radiation safety experience. So this virtual presentation is going to focus more on the fundamentals, and then the asynchronous presentation goes into a bit more of the details for somebody who's already gone through this or very familiar with it. But if you can, I'm going to go through the demonstration piece of this really quickly. Hopefully the audio visual works for me, and then I'll go ahead and share the presentation. but I've got an atomic fireball right here. And then I've got a Geiger-Müller counter. So this is common in the laboratory setting to have, this is a Ludlam Geiger-Müller counter. And in the university setting, it's pretty common to be working with betas or gammas typically. There's a few other radionuclide, it's like PET imaging, for example, but kind of the workhorse has always been betas. And then I have got a plate here for demonstration purposes as well. So I'm going to turn on the audio with my atomic fireball. So the question I would ask the group is my atomic fireball radioactive? If you can hear the GM coming off, it sounds like maybe my atomic fireball is, or maybe it's not. So to get to one of the punchlines, it turned out if you can hear the noises, it's very little. So that's background radiation right now. So good news is my atomic fireball is not radioactive, but is it contaminated? So when I have this plate here, and I put my hand or this atomic fireball on it, have I now contaminated my hand or the candy? So the good news is, if you can hear it, I don't know if you can or not, but anyway, the levels are very low with the with just the candy or with just my hand. So a couple of lessons that we can learn from this is that in this case, the radiation source is fixed. This is Fiestaware plate, so it's naturally occurring uranium that is used for the ceramic. But I can not wear gloves because it's fixed within this. But if I go into the laboratory, and they're working with liquid radioactive material, then I need to be wearing gloves to make sure that I don't spread that contamination, and lab coat and those types of things. So I'm just going to make sure that this is off, and then I will share the presentation and really quick change the audio settings. All right. So now I'm going to go through and talk about the basics here. So some of the questions you might ask is the candy radioactive? Good news, it is not. And you can buy this radioactive candy. Just kidding. You can buy the atomic fireball candy. There's the information there of where to buy it. But anyone that comes to our office, I'm happy to share some. It's very much cinnamon candy. So next I wanted to talk about the public health significance of radiation. So this is an event that occurred in 1987. So it's been quite a while ago, but the importance of learning the lesson from this is the public health significance of if a event were to occur like this today, that it would involve everyone in that community. So what happened in Brazil in 1980s is that there was a cesium teletherapy unit, I believe it was, source, that it was no longer useful to the hospital. The hospital closed down, and they were going through a legal battle, and essentially this teletherapy unit source was left abandoned. Or just, I should say, not completely abandoned, but they just had one individual watching after it. And it got stolen. And unfortunately, as they were using it for scrap metal, as part of taking this apart to sell the lead or the other types of materials, the source, cesium source, about 1,400 curies, was ruptured. And the individuals that received it, the scrap metal yard, they didn't realize that it was radioactive at the time. And they just saw this blue powder. And unfortunately, that individual took it home, shared it with their family, and some individuals passed away from the rupturing of this source. 1,400 curies is a lot. There were also spread of contamination. 54 individuals were hospitalized for radiation exposures, about 250 found contaminated, and 112,000 individuals in Brazil in the 1980s were monitored for radiation contamination. So we definitely, none of us want this type of event to happen again, and there were a lot of lessons learned from this, but also keep in mind, if there were a widespread contamination, there may be a lot of people asked to help. So even if you don't do radiation safety full-time, if something like this were to happen again, there would be lots of people that would be needed to help with that. Okay, so we can move on to going over a couple of basic things that we're going to talk about in this presentation. So I'm going to talk about some of the key characteristics of a radiation safety program. How can we monitor or assess for radiation? Also going to talk about some measures to communicate to upper management. We're going to go over some basic fundamental concepts. I'm also going to define the difference between ionizing and non-ionizing radiation. I want to spend a few minutes to talk about some typical tasks of a radiation safety program, and then also describe how we can monitor for radiation techniques. And then we'll finish at the end with the radiation section of the 50 questions. So what is radiation safety? Well, if we take a industrial hygiene type of approach to define it, radiation safety or health physics is anticipation, recognition, evaluation, and control of radiation hazards. So I'm going to go through and explain each piece of that when we're talking about radiation safety. So what are the hazards? Well, for radiation, we can break it into two main categories when we're thinking about the health effects associated with radiation. So radiation at the high levels can cause acute symptoms for overexposures. So the first thing that you can see clinically is skin reddening or the decrease in the lymphocyte counts, for example. And so I'll go through in a few minutes and talk more about that. But those are some of the health effects that you can see from radiation. Well, radiation is also a carcinogen, but the good news is it's actually a very weak carcinogen. And so I'll talk more about that as well. But radiation has a chance of increasing your radiation risk. I just want to briefly touch on the fact that our regulations are based on the linear non-threshold model. And just put simply, that means that our regulations assume that even a small amount of dose causes a small increased risk. And so whether or not that's true, we could argue about whether or not that's true, but that's essentially how our regulations are formed. And that's why we have the concept of keeping our doses as low as reasonably achievable. So how can we anticipate different sources of radiation on our campuses and our university or academic settings? Well, one thing we need to keep in mind is radiation is naturally occurring. So we have some natural sources of radiation, such as radon. We also have radiation that's used that doesn't require a permit, for example, such as some smoke detectors have americium-241. That's an alpha source that's used to identify the smoke that's in the room. Some exit signs can use up to 10 to 30 curies of tritium. And so that's another example of a source of radiation that doesn't require a permit to possess. And then, like I was showing you, our Fiestaware, if you have antique Fiestaware or some other types of antique uses of radiation, that that also doesn't require a permit to possess. So that's the two that are shown here on the bottom. And then we can also have radiation when certain types of radioactivity, for example, when mixed with a chemical, can cause it to fluoresce, and that's used on watch dials or used in aircraft carrier, for example. So a couple of other examples I have here on the next slide is that we have radiation that's used for some foods to prevent pests from living or prevent or have the food last longer. So that's the food irradiation that's shown here. We also, I'm sure many of us have had dental x-rays in the medical procedures, even bananas. If you know some health physicists or radiation professionals, that all bananas have potassium, and there a portion of the potassium, some of that is naturally occurring radioactive, so that's potassium 40. And so it's not enough to measure with a GM, for example, but it is a very small amount of radiation that we're taking inside of our body. And then if you have industrial radiographers that come on your campus or come across your city, this is the example that's shown in the bottom left, where they would have a source that would come out and they would check on the weldings for that. So there's a lot of different applications of this, and if you want to read more about these different applications, you can go to that radiationanswers.org website. So if we switch and go ahead and talk about common sources of radiation on campus, is we have the research labs. Typically, what they're working with most is betas and then some gammas. And then we also have, for example, diagnostic uses of radioactive material. There are some static eliminators that have very low activities of polonium-210. You may have liquid scintillation counters that have low activity sources associated with that as well. And then there may be radiation producing devices on campus. So for example, if they have x-ray machines for clinical purposes, like dental x-rays or chest x-rays that we're all familiar with, but there also might be x-ray machines used in the research setting as well. For example, electron microscopes, they're no longer required to be registered here in Texas, but that's one example, as well as different applications of x-ray machines on the industrial side as well. And then I just wanted to also briefly mention about lasers. So this presentation is not laser safety, but keep in mind that lasers are may be utilized on your campus as well. And so they may class 3B and 4 lasers, for example, and they might be benchtop or they may be confined. So lasers come in a lot of different applications for that as well. And then you might have electric and magnetic fields. And so, for example, if your university is working with MRI or NMR machines, as well as considerations for other types of electric and magnetic fields. So how can we identify radiation on our campus? So if it's regulated, it requires labeling. And so the most common here for radioactive material is we're going to have this caution radioactive material trifoil. And it's typically going to be either in red, black, magenta, have that symbol that's shown there. And then for radiation producing devices or x-ray machines in Texas, that labeling is required as well to say caution radiation area or caution x-ray, for example. And then in shipments, there's going to be requirements depending upon the levels to have the different labeling for the white 1, yellow 2, yellow 3 type of examples. And so We can identify radiation through the labeling. And the good news is we can also identify radiation through radiation detection equipment. So probably the most common in the university setting would be the Geiger-Muller counter, which is what I was showing you there. And so there's two examples here, would be a pancake probe, which just looks like a small pancake. And then on the right here is also a Geiger-Muller counter, just in a different shape. So for evaluating the hazards, we can monitor it with different radiation detectors. And then we can also assess for removable contamination, as well as provide dosimetry. So when we talk about assessing the removable contamination, we can use the small little filters that are shown here, or even at our facility, we use cotton swabs that just look like routine Q-tips. And we're basically just wiping that area and then running it on our liquid scintillation counter. Or if you're out in the lab, you could wipe the area and run it on your GM or Geiger-Muller counter, for example. And then I've got some dosimetry examples that are shown here for the different types of badges that could be provided, whether it's full body or a ring badge. Those are some examples there. So how are we controlling the radiation hazards? Well, in general, we're typically issued a permit. And so I would say I'm very Texas focused, but we give this presentation, when we give it, we have folks from all across the United States. And so if we think big picture wise, we have the Nuclear Regulatory Commission that issues the radiation control permit. And then you can have the radiation control rules and permits through the Nuclear Regulatory Commission, or you can have what are agreement states. So here in Texas, and I think at this point, over 40 other states, maybe more like 45, have agreement states that agree to follow at least the Nuclear Regulatory Commission's regulations. And so here in Texas, we have the Texas Department of State Health Services. And so we are issued permits under Texas Department of State Health Services radiation control. And so they issue permits for radioactive material license or for a radiation producing device. And one note I wanted to point out here is that in the longer version, or the asynchronous, I have some links on, if you're not in Texas, where to find out which agreement, which state you follow. So you can go to the NRC's website that I have listed there, or you can go to the CRCPD's website. Another thing I wanted to briefly mention here as well is that the X-ray and laser regulations vary a lot by state. So I'm very familiar with Texas regulations, but in other states, they may not have as robust of regulations or program. So definitely on the X-ray and laser side, that level of oversight may vary by state. So as part of the permit, you're required to have a radiation safety officer. So each permit has that officer. And then if you have a broad license for radioactive material, there's a requirement for a radiation safety committee. So I need to pause here and briefly describe the difference between a broad license and a specific license. So for radioactive material, if you have a specific license, then it will say you're authorized for these specific radiation items. So as we get further in the presentation, I'll show you a permit example that shows you what you're authorized for. But if you have a broad license, you're issued a bigger blank check, so to speak, in that your committee takes on some of that oversight. So there's still oversight with the state, but your committee allows the researchers to, or the clinical authorized users to work with a wide variety of radionuclides so that there's less correspondence with the state on a day-to-day basis. So that broad license allows you that ability to do more changes in-house. And so the fundamental purposes of the radiation safety program as part of this is to maintain doses as low as reasonably achievable. So what are the requirements of a radiation safety officer or the requirements of the radiation safety program if you have a pretty big staff? Well, they're actually listed in the regulations. So what I'm showing you here are for radioactive material use in the state of Texas. If you're from another state, I'm sure that your regulations will be listed there for the minimum duties as well. But it's typical things such as training. There may be dosimetry that's required, reporting of events, responding to emergencies, and keeping up with permit changes, for example. And then probably the last one here is every facility is going to have BS. The radiation safety officer is going to be that primary point of contact in dealing with that type of radiation permit. So what does a radiation safety officer have to follow, or those individuals working with it? Well, of course, we have to follow the rules and regulations. So that's in Texas. It's typically listed on your permit, what rules and regulations you have to follow. So for example, in Texas, they're broken down by type. Human use of radioactive material versus veterinary use of X-ray machines, and human or veterinary. There may be industrial applications for the different permits. So those are listed. You also have certain permit conditions. So after they list what you're authorized for, so for example, this has Americium 241 is the first item. So on this fake permit here, they're authorized for a sealed source and the activity not to exceed 1 curie of Americium 241. Well, as we get further down on the number of pages, there'll be permit conditions. For example, one for us is if we have researchers working with over 100 millicuries of tritium, we have to do weekly urine bioassays. So that would be a permit condition. And then you also have tie downs, which is typically going to be on the last page, is letter correspondence that says what you have to follow as part of this permit. And so if you're managing a program with that type of license or registration, you want to make sure that you have a copy of all of those tie-down letters. Okay, in the interest of time, I'm going to continue to move on to the basic radiation safety concepts. So I wanted to talk about the difference between radiation and radioactivity. So put simply, with radiation, it's the emission of energy, and it can be in the form of waves, such as gamma rays or photons. And then it can also be in the form of particles, such as alphas or betas. So that's the generic version of what is radiation. Well, the next thing to define is what is radioactivity. Radioactivity is the spontaneous emission of radiation from the nucleus of an atom. So if we think about something that's naturally occurring, radioactivity, for example, uranium, or we can think about the banana example where we have potassium-40. Those are radioactive or spontaneously emitting the radiation. And then we've got an image shown here is just a simple dental X-ray machine. So that is something that is a radiation producing device. So that one, once we unplug it, we cannot produce radiation from it. So it's a big difference when we're talking about how we manage those two types of radiation. And it also makes a big difference if there's an emergency and we think about how we respond to the radiation. Is it radioactive or is it just a radiation producing device? Another thing to define is the electromagnetic spectrum and defining whether or not it is ionizing radiation or non-ionizing. So for ionizing radiation, put simply, it has enough energy to produce ions. And so the levels are, I think, around 60 to 70 kiloelectron volts to produce ionizing radiation. Basically, you don't have to remember that particular energy level. What you need to just remember is that typically betas and gammas and alpha rays, those have enough energy to cause ionizations in, for example, the human body. And so that's why we term that ionizing radiation. If it's non-ionizing, it cannot produce ions in the body. So I think that this next slide can maybe help describe it a little bit better, is the electromagnetic spectrum. So we can define things in terms of their energy or their wavelength. And if we've got kind of in the middle of this slide here, we've got the wavelengths. And so gamma rays and X-rays are ionizing radiation. So they have sufficient energy to cause those ionization events. But we have UV, visible, and infrared and radio waves are all examples of non-ionizing radiation. So technically, visible light that we all see is a form of non-ionizing radiation. But we typically don't describe it in that way, but we can describe the different visible light spectrum by the wavelength. So I want to move on and talk about some basic nomenclature when talking about radiation. So the X here, if we remember from chemistry, this is our elements. And so I've got two examples, tritium, which would be hydrogen, and then P, the P would be for phosphorus. So the X identifies the element, the Z that's shown here is the atomic number or the number of protons. And so the number of protons always identifies what element we have. And then the A is the mass number. So it's the number of protons plus the number of neutrons. So when we're talking about radionuclides, we typically leave off the Z that's shown there because it's understood because it identifies the element. But for tritium, we can just do some simple math to see that the number of neutrons for tritium or hydrogen 3 is 3 minus 1, so that would be 2 neutrons. Or we could do the same thing for the phosphorus 32. So I just also want to briefly mention the chart of the nuclides. I have a desktop version here, but what we've got on the slides here is the all of the radionuclides are shown, and the black here is anything that is stable. So the chart shows you both things that are stable as well as things that are radioactive. So anything that is yellow or green on this big chart would be anything that is radioactive. And then the black here means that if it's radioactive, it's emitting something to become more stable. So that's kind of the big picture of this chart. And then if we zoom in from this chart, we can look at things such as the half-life of their radioactivity and then what it emits. And so We got the cesium-132 has a 6.5 day half-life is how we would read this chart. So the next thing I wanted to talk about is radioactive decay. So if it's radiation is radioactive, it's decaying to not to disappear, but to something that is more stable. So in the example of americium-241, it's emitting an alpha particle, and with that emission of the alpha particle, it's actually transforming into neptunium-237. So if we know one of these, or two of these, I should say, then we can figure out what it's decaying to. So with americium-241, if we know it decays by alpha emission, we can just do 95 minus 2 is equal to 93. 241 minus 237, 241 minus 4 equals 237. And so it decays into that neptunium-237. So when we have that radioactive decay, we have to use a term called the half-life. So the half-life is the amount of time required for half of the original amount to decay. So we can graph the half-life over time. And with this example here, we would start with the half-life of 15 minutes, for example, and then we can go through. So 2 half-lives would be 30 minutes and continue on. And so we can calculate it or we can view it graphically. We can, when we calculate, we can use this formula. There's actually a couple of different formulas out there that we can utilize. So our activity is equal to our original activity. So this original activity would be A naught, E to the minus decay constant, that lambda times time. So that decay constant is actually equal to the natural log of 2 divided by the half-life times that time. So there's actually a easier way that we can do this. We can, go to a website, or we can do just rough calculations. So if anybody wants more details on this, I will definitely be around at the end for it, or happy to point somebody towards some good resources for that. But that's the basics of how we can calculate that. For the different types of radiations, I would say that the most common in the laboratory setting would be the betas and gammas, but we'll talk about the alphas, betas, gammas, and neutrons when we think about how can we protect ourselves against these different types of radiation. So an alpha particle is a helium nucleus. It's if we compare it to betas and gammas, an alpha is very big compared to the beta and gamma, because betas are just electrons, for example. And so it's typically has a higher energy that's associated with it, but because it's so big, it's easier to stop or to slow down. And so it has typically the lowest amount of penetrability, but it's a very much a concern if it's an internal hazard. So for an alpha, it can't penetrate typically the dead layer of the skin, but if we inhale it or ingest it, we have a big problem that we have to deal with. So for betas, it's an electron, and it's typically emitted in a spectrum of energies, and it's typically shielded with plexiglass. So here in a minute, I'm going to move to the next slide. But that's typically what it's shielded with. And then we have gammas that are photons Those have the highest amount of penetrability, and those can typically travel the farthest distance. And then neutrons requires specific shielding requirements with that, and they also are slowing down. And so they're just a little bit more complicated to talk about on an introductory type of presentation. So for the alphas, this gives us a picture to show graphically that alphas generally have high energy, but they're easy to stop. So that's why we say that we can shield them with a piece of paper because they typically can't penetrate the dead layer of our skin. With betas, they're can travel further distances, and they're kind of in between on the distance traveled between betas and gammas. We typically shield with plexiglass with betas because if we have high energy betas and we shield with a high density material such as lead, we can produce X-rays. And then gammas are typically shielded with lead. So how do we minimize our radiation doses? Well, this is probably a good slide to keep in mind is we minimize it by using the ALARA concept, keeping our doses as low as reasonably achievable. So we can do it by reducing the time we're spent near a radionuclide, and we can also increase the distance, or we can add appropriate shielding. There's some others that are there, such as using less as well. But I wanted to briefly mention about the distance is it follows the inverse square law. So if we think about our dose, if we start with one dose of 1 Roentgen per hour, for example, and we go back a distance of 3 feet from 1 foot to 3 foot, instead of reducing that dose by a third, for example, we're actually reducing it by 1 squared over 3 squared, which turns out to be 1/9. So distance is a very effective way to reduce our radiation dose. And then the shielding is it needs to be appropriate for the type of radiation we have. So in this case, we have some examples of shielding the primary vial with plexiglass, for example, or we can shield the work area with lead bricks or where they're working with that area. So I wanted to briefly mention about brimstalling. So brimstalling is when we have something that is a higher energy beta, such as phosphorus 32, and we add a dense material with that, such as lead, then we're going to produce X-rays off of that. So I also wanted to spend a few minutes talking about the units when it comes to radiation or radioactivity. So when we say the radioactivity, that's a measure of the rate of decay of something that we have. And so it's a measure of how much of that radioactivity do we have. And that's in units of curies or becquerels. So 1 curie is equal to 3.7 times 10 to the 10th becquerels is the conversion. I would say if you're not working with radiation routinely, all you really need to remember is that a curie would be a large amount of something, and a becquerel is 1 transformation per second, or a small amount. So if someone says we have a spill of 100 curies of radioactivity, then I would be very concerned and ask a lot of questions. But if someone said we had a spill of 100 becquerels of radioactivity, that's not that much radiation or radioactivity. So that's what I would say to folks if you're not working with it on a routine basis. Next, we can think about how much charge is produced in air. So we have here in the US a term for that called exposure. That can be in units of Roentgen. And so we have some detectors that measure in the units of exposure. Well, we're interested more in how much energy is deposited in the material or radiation absorbed dose. So that is, here in the US, is units of rads, or internationally would be gray. But as I was talking about earlier, an alpha is a lot more damaging, so we have to have a term called dose equivalent to be able to equate those. And so that dose equivalent is in units of rems or sieverts. So the US terminology would be that those rems, and that's what our dose limits or regulations are based off of, is how we equate those. So what do we really need to know? Well, for betas and gammas, 1 Rankine is approximately 1 rad, and it is equal with that quality factor of 1 rem. For alphas, neutrons, and protons, we simply need to know that 1 rad does not equal 1 rem. And so we have to take into consideration the quality factor. For alphas, it's 20. And so we can see that it's more damaging if we have the same dose of radiation for an alpha. So radiation can cause biological damage. It can cause deposit energy in the body, and the damage really depends on the dose, just like with chemicals, and whether it's the DNA damage in the body or ionizations. These can lead to free radicals that can cause damage as well. So if we think about the different health effects, we really need to break that into 2 groups: the chronic exposures or acute exposures. So when we think of chronic, we're typically thinking of low doses of radiation over a long period of time. When we think of acute health effects, we're thinking of someone that's gotten a higher dose over a shorter period of time. And typically that threshold for seeing acute health effects is 10 rem. And so examples are there: skin reddening or cataract and those types of things. So when we think of the very high end of radiation exposures, we can break it down into the hematopoietic, gastrointestinal, and central nerve systems. So for the health effects of 100 to 700 rad, this is, just to put this in perspective, this is well above the background limit there. Well above the background limit and the occupational dose limit. And then the occupational dose limit, for example, is 5 rem. And so that's that 100 to 700 rad or rem is well above that. And then we've also got the gastrointestinal problems. If we have a very extreme high dose of radiation, it can even cause central nervous system failure. And so that is listed there. Thank goodness none of us should be having to deal with these very high levels of radiation, but it's something I did want to mention. I also wanted to mention chronic effects of radiation. And so in the early 1900s, we had radium watch-style painters, and these individuals were working with radium. They were getting repeated small doses of radiation. And so they were actually painting with a very fine tip to their paintbrushes, the different watch styles or other types of materials that you see here. And to get a fine tip, they were actually putting the paintbrush in their mouth and twisting it. And unfortunately, they learned the hard way that radiation can cause health effects from those repeated small exposures. And radium is a bone seeker. And so they developed a lot of problems with the jawbone as well as their bones from these repeated small doses. And so fast forward many years later is that we now have the Nuclear Regulatory Commission, the OSHA, and those types of things so that we can address the concerns with radiation. So I wanted to switch gears and talk about radiation safety metrics and measures to communicate with upper management. So the good news is with radiation safety that the number of deaths associated with radiation is extremely rare, and the number of injuries and illnesses are rare as well. And so in the absence of those, we also want to look at what are some other measures that we can communicate to our radiation safety committee or management or the like. So we do get routinely inspected for radioactive material and x-rays. And so they do issue fines. I would say if they don't issue a fine routinely, our experience is it's not routinely that fines are issued. They're really reserved for gross violations, but we're definitely routinely inspected. And for us, for example, why we show all these metrics and measures is that we're not going to, the best that we can get is a letter that says we appear to be in compliance at the time of our inspection. So anyway, I wanted to keep us on time as well, is we want to report not only the state inspections, but how we do from our different safety audits that we do internally. So we have the radioactive material inventory that's shown, our inspections internally as well as from the state. We also issue dosimetry, so that's included in our monthly numbers as well as our training. And then we also have in our monthly numbers how many devices that were inspected and the like. Okay, so I'm going to move on and talk about the mission of radiation safety. So we assist all levels of management in providing and protecting the workers, the general public, and the patients, and working with these radiation hazards. And put simply, we want to make sure that folks are as healthy as when they go home as when they arrived, ideally. And then we have lots of regulations to comply with associated with that as well. So some of the different aspects of a radiation safety program include an ALARA program, so keeping our doses as low as reasonably achievable. We're also where the bulk of the radioactivity is received at our facility, so we're involved with both the shipment and receipt of radioactive material. We're involved with radioactive material or radiation producing devices inventory tracking. There's always training. There's security associated with radioactive material. There's requirements for silt source leak test, bioassays. There's concern about the declared pregnancy program and how we address the workers working with that, as well as environmental monitoring, guidance, and oversight. So for training, the minimum requirements for training is listed in the regulations. And so here in Texas, that would be the regulations that's listed there. And I wanted to mention specifically about this is also the requirements for how long or how extensive the training needs to be really varies by institution and permit, because, for example, if they're just working with microcurie amounts of radioactivity, the training may be very short, versus if they're working with 100, 200, or 300 millicuries or curie amounts of training, the training may be more robust. And so that's why you see a wide variety of training from institution to institution of how long the training is, as well as how robust that test is. So we can survey for contamination with our different detectors. So there's some examples that are shown here, and the filter paper where we can wipe the area and make a map of the room, for example. And then we also need to know how to clean up a spill when we're talking about loose radioactivity. So I've got an example here where we would spray the area and wipe from the outside in, making sure to have appropriate personal protective equipment and putting that into a radioactive waste container. And then we can survey the area after we're done to make sure that we've cleaned up all of the spills. And then on the high end associated with that, there might be notifications that are required to our permitting agencies. So how would we clean up a spill? We can clean up that spill with the procedures that were listed previously there. And then we also want to do that post-decon. And then we also want to have an incident investigation report. So if it's something minor, very minor spill, then we don't have to contact our regulatory agency. But if it were major, we would need to contact the agency. And then bioassays might be a consideration as well, depending upon what the individuals were working with, the radionuclides and the amounts. So also we need to consider emergency response applications. So if some of us are associated with a hospital, how would they handle the patients that are coming in? A very good question to ask is, was that individual contaminated or were they just irradiated with the radiation? Because that makes a big difference in how you handle the treatment of that patient. So that's why it's important to be able to monitor the workers for contamination as well as the patient. So if we think about our dosimetry, how we can assess that is the regulations say that we are required to monitor anyone who's likely to exceed 10% of the dose limit. And so I would say at our facility, we probably are generous with our monitoring is how I would phrase that, but that we monitor those that would like the badge typically, as well as those that are over that 10% occupational dose limit. So we've got an example of a whole body badge and a ring badge that are shown here. Something to keep in mind is that there's the US average annual dose. So anything that you're monitoring for, if I can go back here really fast, any doses that we get from this, that is above our background sources of radiation. So it is taken out the doses that you get from natural sources of radiation, which the largest contributor here would be radon, or medical procedures that you would have as a patient. So currently, the estimate of the US average is about 625 millirem that's shown here. And by far, the largest piece of that is the medical. I'm sorry, the medical is the largest that is not man-made. And so CT would be the highest here. And then we've got for a breakdown, whether it's for fluoro or diagnostic imaging. So this one is probably a good one to mentally flag, is that we have exposure limits. And so the one, if I was only going to remember one of these, is the whole body annual exposure limit of 5 rem per year, or 5,000 millirem. And then because certain organs are more radiosensitive, we also have organ limits. So we have an eye dose that is currently at 15 rem, or 15,000 millirem per year. And then because the fetus is very radiosensitive, and because children, for example, are more radiosensitive, we have a lower dose limit, and that is 10% of the occupational limit for others. So if we just look at the whole body, that would be 500 millirem over the gestation period, or 9 months, and then or 500 millirem for a minor, which would be someone under the age of 18. We also have general public limits to keep below 2 millirem per hour, or 100 millirem in a year. So who needs the dosimetry? It would be those that are considered likely to exceed 10% of their annual dose limit. So I've already talked about several of these, but for the declared pregnancy program, we want to minimize their doses just like with any others. And we really use it as an opportunity to educate the individuals if they're not very familiar with it. Working with all the radiation, we use this as to give them peace of mind, because we find that most of our pregnant workers are actually working at very low radiation levels for them. And so it is up to the individual to declare. So we provide training for everyone, and then they can reach out. We give them multiple ways to reach out to us so that they can actually declare their pregnancy, to have that lower dose limit, and to give them the knowledge of ways to reduce their exposure. So here are some typical things in the radiation laboratory. So we've got the caution signs, we've got the worker that is working with the radioactive material. They're going to have where they're storing their radioactivity with a label. And then I've got a couple of examples of liquid scintillation counters where they're assessing the radiation. So just to generically speak about radiation detection, is that Geiger-Müller counter, for example, is counting the ionization events that occurs in that radiation detector and getting the counts associated with that. So I've got some examples shown below here of different types of Geiger-Müller counters. But to break it down into different classes, is we have the gas-filled radiation detectors, a Geiger-Müller ion or proportional counter, and then we can also have liquid or solid detectors. So we can have where we're monitoring for loose contamination on the benchtop, or for example, checking our feet. So the GMs are a common example associated with that. And then to briefly mention, if you see this ball shape here, that would be for a neutron detection capabilities typically. So if we talk about liquid scintillation counting, a lot of times if we're looking for removable contamination, we're going to use that cotton swab. We're going to add it to our container with our cocktail and get the counts off of that for the radiation levels in that laboratory. Those liquid scintillation counters are typically expensive, so not every place is going to have those, but definitely many places often have the Geiger-Müller counters, and they come in different sizes. And then we've also got an example here of taking the waste to a landfill. Is many of those landfills have sodium iodide detectors that are pretty low levels to detect the radiation coming off of that. And then we can also have pocket ion chambers to detect the ionizations or levels off of what we're working with the radiation. So I have a question here. A portable Geiger-Müller counter cannot detect, is it A, phosphorus 32, B, chromium 51, C, rubidium 86, or D, tritium? And considering this is virtual, I will go ahead and answer my question. It cannot detect tritium. I'm sure some of you might have put the right answer in the chat, but the reason why it can't detect tritium, or that H3, is because it's a very low energy beta. The beta associated with tritium is only 18.6 keV, and because it's such a low energy, it can't beta can't get into the GM detection area. And so the question is, how would you detect it? Well, at our facility, we have to do wipes and run those wipes off of a liquid scintillation counter. So that's one of those that we want to make sure that our researchers know, especially those that are wanting to work with tritium. Okay, so thank you for everyone that, if you did answer in the chat. So I was told to cover this a little bit quicker so that we have time in case there's questions. So I'm going to finish up with the 50 questions. And I did have a view of the Texas Medical Center. For those of you that aren't familiar with Houston, or the Texas Medical Center is our offices right next to this taller, shiny building. I'm not sure if you can see my pointer, but the 50 questions here that we're going to go through and talk about is the annual dose, the related to dosimetry. So do I have Scott here that can share those 50 questions? Don't actually have the 50 questions right in front of me, but I will go ahead and generically talk about them. And then hopefully we have someone that can pull those up. Is what is the annual dose limit? So that annual dose limit for occupational employees is 5,000 millirem or 5 rem in a year, and that's for the whole body dose. So the next question is probably what is required for the issuance of a dosimeter, and that is you're required to issue a badge for anyone who is likely to exceed 10% of the dose regulations. And then we at our institution badge more individuals than that. And then the next question is, what's the difference between radiation and radioactivity? So radiation is the emission of energy, and it can be in the form of particles or gamma rays, but radioactivity is spontaneous emission of radiation. And then the next one was, what's the difference between something that's radioactive versus something that's contaminated? So if it's radioactive, it's spontaneously emitting radiation. And then if it's contaminated, then that means we've gotten radiation where we don't want it. So we've gotten it on our skin, we've gotten it on the benchtop, on our clothing. So that's, put simply, that's what radiation contamination is. And so when we're dealing with it in the laboratory setting or emergency response setting, we need to know what is the difference between something that's radioactive versus contaminated. So this is radioactive, but it is not contaminated because it's fixed. So that means I can touch it, but I can't spread it unless I break it into little pieces. So this would be a good example of something that's not contaminated, but it is radioactive. So what's the predominant emission type used in the laboratory setting? So for us at UT Health, that would be tritium. I'm sorry, I'm answering the next one. For us, it would be betas and gammas. So betas would be phosphorus 32. And so that would be S35 would be another example. So phosphorus 32 is a higher energy beta. Sulfur 35 and C14 are medium level beta energies. And then tritium would be a common example of the radionuclides at those low levels. And then how do we detect tritium? I've already answered that one as well. That would be with the liquid scintillation counter. And it's important to know that we can't detect tritium with a Geiger-Müller detector that's shown here. And then we have what is a broad license, and that broad license is, I can maybe go back to sharing. That one allows us the ability to have a permit in-house where we have a lot more flexibility so we don't have to correspond with the state again. So let me go back and share these slides. So this would be an example of that radioactive material license. And then the broad piece of that would be, we would have, for example, the C is showing any radiation from atomic number that is up to atomic number 84 or greater. Okay. So excuse me, considering there's some time, I can stop and see if there's any questions.
And just for those in the course, the 50 questions are available on Canvas. It's under week 1, and it's going to be under the asynchronous and additional resources. If you go down to additional resources for week 1, there's a link for 50 questions worksheet without answers and the 50 questions worksheet with answers. So does anyone have any questions for Janet or Robert Plant?
Well, thank you everyone who stayed on till the very end.
That's it. We'd like to thank all of you for attending our academy. Thank you for setting time 3 consecutive Mondays. I hope we got a lot of useful information out of this. I hope you got stuff that you can apply to your work environment right away. Please fill out our evaluations. We would love to get feedback to know how we can improve and things that you've learned most out of this course. Again, if there are any questions, last call for questions. A lot of thank yous in the chat, so we appreciate those. Thank you, everyone, and have a good afternoon.